All right. So, good afternoon, everyone. Okay. Hope you had a great lunch. As you have fed your body, we'll now try to feed your mind. Okay. So, welcome to the third and final day of GBCon 2021, Genomics and Bioinformatics Conference, Overcoming Challenges and Building Opportunities. Okay. I'll be your host for today. Okay. I'm John Michael C. Yap, Program Director of the Core Facility for Bioinformatics of the UP Philippine Genome Center. Okay. So for this day, our theme will be overcoming challenges and building opportunities in biodiversity, ethnicity, and forensics. And to kick off our activities for today, I would like to welcome the keynote speaker for today's proceedings. Okay. Um, She's the director of the Biodiversity, Ethnicity, and Forensics program of the Philippine Genome Center. Help me welcome to the floor Dr. Maria Corazon de Ungria. Ma'am Cora, the floor
Hello,
is
good
now yours.
afternoon. Good afternoon, Dr. Yap. Okay, so we can start. Can you see?
Yes, ma'am.
All right, okay, so let's start. Well, first of all, good day. The title of my talk as shown here is Biodiversity, Ethnicity, and Forensic Genomics, Challenges and Opportunities. I have used genomics as part of the title because of its wider scope compared to genetics. Genetics is merely the study of genes and their roles in inheritance. On the other hand, genomics describes the study of an organism's genes with each other or with other organisms and with the environment. Hence, genomics does not end with a person or individual organism, but includes the effects of the person or organism's existence on others and the environment. Following this definition, forensic genomics is the application of genomics to answer questions posed by members of the legal profession as part of a criminal investigation or civil action. I've summarized on this slide the program challenges during the pandemic. Firstly, there was a reduced access to the laboratory, offices, library, and the internet. Reduced access to our colleagues, which decreased the amount of interaction and support that we could give each other. Delays in the release of money, including salaries, and changes in research funding for our projects. Secondly, we also had to adopt non-traditional work-from-home arrangements, which removed the divide between work and home, creating chaos compounded by unstable and intermittent communication channels. And lastly, the need to remind one another that we should accept ourselves for what we can and cannot do. That at the end of a very long day or a very long night, still finding our task far from where they should be, we should pat ourselves on the back or even embrace ourselves because we did our best, given all that we have and all that is happening around us. Although these challenges could be discouraging, it has also enabled us to tap our creativity and personal resilience, to open doors and to discover new ways to achieve the same goals. The challenges we face can wake up our survival instincts to embrace these challenges and make the most of what we have, while learning to appreciate those we took for granted in the past. The presentations of our colleagues in the program today will show us specific examples of creativity and resilience, which I hope will inspire all of us and perhaps help us reach out and collaborate. I am reminded of the story of the stick, which I learned when I was very young. It is always easier to break one stick than to break a bundle. And I hope in this program, we work as a bundle. This slide summarizes the applications of genomic studies in humans, livestock and wildlife, plants, insects, fungi, bacteria, and viruses. Now let us briefly go through the topics covered in today's seminar, or to be covered in today's seminar. The first speaker, Mr. Frederick Delphin, focuses his research on human population genetics. Fred will tell you about the extreme value of understanding the genomic variation across Philippine populations, including our indigenous brothers and sisters. However, COVID came when his team was already preparing to do field work to collect saliva samples from volunteers in different regional centers. In addition, because of the relative novelty of human genomic research in many areas, it is interesting to learn how the team reached out and used different communication channels to inform policymakers, government institutions, universities, and communities. To meet project goals with the time frame, the team tapped ongoing research projects that already had collected biological samples. So Fred will give you all this information. The next speaker, Dr. Lara Espada Murao, will talk about her work as director of PGC Mindanao. In fact, PGC Mindanao is the first regional PGC, which was able to expand and acquire infrastructure amidst a pandemic. In fact, Dr. Lara's team played a significant role in training and testing SARS-CoV-2 variants in Mindanao. PGC Mindanao in Davao was launched in 2019, followed by PGC Visayas in Tacloban the year after. The establishment of regional DNA laboratories and the decentralization of DNA testing are welcome developments that should accelerate the support that PGC can provide local government units, increasing regional capacities for genomic research. In addition, Dr. Lara will tell us about her exciting research on fruit bats, opening more doors for the rest of us to learn about the diversity of flora and fauna in southern Philippines. The third speaker, Dr. Din Matias. Din, I don't have your photo. Dr. Din Matias will be talking about the challenges of studying population genetics in the Philippines, given 1, the need for computational biologists across all fields, and 2, the insufficiency of computational infrastructure in many areas. Knowledge of the diversity of different taxonomic units is critical in formulating strategic conservation efforts amidst the continued rise in the Philippine population. The intrusion of non-Philippine populations, and you know who they are, in our seas and land, and the effect of climate change. Our last speaker, Dr. Ian Fontanilla, will discuss our efforts to initiate the Earth Biogenome Philippines Network for conservation, food security, and therapeutics. Through the program on biodiversity, ethnicity, and forensics, we are actively forming a consortium of researchers inside UP and outside of UP. This group will work together to share the best protocols, prioritize research efforts to select the more important taxonomic units, and formulate policies for data access and benefit sharing, following relevant international treatises. I also would like to acknowledge Ms. Jaslyn Salvador and Ms. Gavelyn Calakal. They are not speaking today, but they have continued to work in forensic genomics, as shown in the respective papers that are already published. Jaslyn's interest is in population studies of forensically relevant markers. Gavelyn, on the other hand, is our bone DNA expert, whose work will impact the analysis of human remains post-disaster, also of victims of extrajudicial killings. And we hope that that will be coming soon. We also explored ways to use DNA in animal forensics during the lockdown. The paper entitled Incorporating Animal Forensics in Routine Meat Inspection in the Philippines reviewed different protocols used for determining the identity of animal meat that are sold as food, including those that are sold in halal markets. 3 days ago, I've discovered that this paper was cited on several online news platforms. We hope, and we've reached out to the National Meat Inspection Service, that they would consider the recommendations made in the paper and for other groups to pursue more research in the area. Because of the events in 2021, the program studied human genomic and genetic research governance in the Philippines. 3 critical areas were identified. First, the need to review human biobanking policies and data sharing in the Philippines, understanding the requirements that are made when we publish papers in journals, as well as the social consciousness of the free and prior informed consent provided by the participants. Second, the ethical challenges in the establishment of forensic DNA databases in Southeast Asia, following the use of forensic genetic genealogical testing and public databases in the US to generate investigative leads. And third, the ethical challenges in genetic research with indigenous cultural communities in the Philippines that came out of the work of foreign researchers who did not comply with local, ethical, and institutional policies in the Philippines. And to end, this slide answers the question of why study and sequence life. First, to revolutionize our understanding of biology and conservation. Second, to conserve and restore biodiversity. And third, to create benefits for society and human welfare, ensuring that excellent, rigorous, and ethical science be used as the foundation of genomic science now and for future generations. In the program on biodiversity, ethnicity, and forensics, we hope that more people will collaborate, cooperate, and communicate with us such that we can do more science for the service of Philippine society. Thank you and stay safe. Oops. That was
All right.
a picture, sorry. Thank you.
All right. Thank you very much, Ma'am Cora, for that keynote speech. I hope that it definitely whet my appetite on the next couple of talks that will be given by our guest speakers, right? And I'm actually giddy to see or hear what will be given, what insights will be given by our speakers. So giddy, in fact, that I forgot a while ago to tell our audience that we are actually streaming live on Facebook. Okay, if you could visit our Facebook live stream page at facebook.com/phgenome. The link is also can also be found in our chat in the chat box right there. Okay. Also, I would like to remind everyone that you could have some degree of interaction with our speakers, including Ma'am Cora, who's just given her keynote speech. I'm sorry about that. Okay, so you may submit your questions in the Q&A tab during our speakers' presentations and even during the break later on. The tab is usually located at the bottom part of your screen. All right, so if you have questions or comments, do fire away. Our presenters and our speakers will be very much appreciative because of it. All right. Okay, so now, without further ado, let me now introduce our first plenary speaker. Okay, so our first speaker is Mr. Frederick Delfin from the Natural Sciences Research Institute of the University of the Philippines, Diliman. Mr. Delfin's bachelor's degree in biology was from the University of the Philippines in Baguio. University of the Philippines, Baguio, and his Master of Science degree was from the Molecular Biology and Biotechnology program, now the National Institute of Molecular Biology and Biotechnology of the University of the Philippines, Diliman. Okay, his doctoral fellowship training was through the International Max Planck Research School for Human Origins at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, and through the Leiden University Medical Center in the Netherlands. Mr. Delfin's research focuses on human population genetics, evolutionary genetics, and molecular anthropology. Very interesting, right? So Mr. Delfin leads one of two human population-based genomics projects under the Filipino Genomes Research Program at the DNA Analysis Laboratory of the Natural Sciences Research Institute in UP Diliman. Okay. So without further ado, I now present to you Mr. Frederick Delfin, who will be presenting his talk on human population-based research in a pandemic, engagement, saliva, etc. So, Mr. Delfin, the floor is now yours.
Good afternoon, everyone. First off, thank you to the PGC for this opportunity to share and contribute to this activity. In line with this activity's theme, the title of the talk is Human Population-Based Research in a Pandemic, Engagement, Saliva, etc. This is just the start of the talk. However, credit should be given to many institutions, individuals, and various Filipino groups who have helped us through the years, without whom we would not have the research background and a platform for a Filipino genomes research program. The FGRP is funded by the PCHRD and is currently composed of 3 projects headed by Jazelyn Salvador, yours truly, and the head of our lab, Dr. De Ungria. Here is our FGRP cast and crew with a couple of our PCHRD program project managers. From left to right, we just call the projects FGRP Project 1, 2, and 3. Of the 3 projects, projects 1 and 2 are the population-based projects. We call these Phase 1 projects because they include only 41 groups. This is in consideration of the overall research costs of having to include all Filipino groups. We will work towards succeeding phases where we get to include other Filipino groups. So for FGRP 1 and 2, you have again the cast and crew and the 17 RC groups and 24 EL groups considered in Phase 1. There is a framework for basic research on human population genetic variation. Inside the dashed line are the stages of the projects. Outside the dashed line are details relevant to research involving human participants, vulnerable groups, and in particular human genome data. Guided by community-based participatory research, iterative dynamic processes, local laws, guidelines, and issuances, consider the dashed line as the relationship of partners from funder to participants from the start to the end and beyond the research process. At the social preparation stage, we partnered with different academic institutions to help represent the 17 RC groups. FGRP staff were delegated to oversee different regions. Pre-COVID, we were able to visit some of our AI partners to hold inception meetings and to start up research agreements. But because of this infectious agent, virtual methods were maximized. Because of efforts pre-COVID and during COVID, various research agreements were forged between institutions. The picture shows a pre-COVID on-site MOA signing between institutions, facilitated by FGRP1 project leader Jazz, together with Cian and May. Still on social preparation, we developed a research communications and ethics instrument, realized here as version 1 and version 2. Exemplified here by version 1, the RCEI can help facilitate the appreciation of the research process and research ethics. Version 1 is popularized into version 2, where you can see FGRP researchers Gino and Dina talking to potential partner participants Juana and Juan. You can see Juan ask, and Dina tells him about DNA. Juan and Juana are told about pre-FGRP Filipino population genetic studies. Juana is told about sample processing, from saliva to DNA, DNA to a biobank, and from DNA to DNA sequencing, and the DNA data to a data bank. Version 2 has been and is still being translated into several Filipino languages for dissemination. And how do we use the RCEI? As depicted here, think storybook reading. Though participants would have to be more physically distant, this can be facilitated by each participant having their own copy. We just have to make sure that everybody is on the same page. And again, because of COVID, we can have virtual RCI reading sessions, as exemplified here by one of several RCI reading sessions we've already had with our partner AIs. With its evolving versions, the RCEI is meant to be carried from the start through to the end and beyond the research process. It can serve as a template for stakeholder input, and for the partner participant, it is proof that they participated in the FGRP research process. There are other research communication materials such as posters with virtual links to brochures. The examples here are in English, Filipino, and Ilocano, preferred for information dissemination by our partner AI in San Fernando, La Union, Don Mariano Marcos State University. These materials can be found in the DNA Analysis Laboratory Facebook account. I invite all of you to check them out, perhaps share them as well, and perhaps if you find yourselves qualified based on our inclusion criteria, you could sign up as a potential partner participant. Now, to sample collection. Because of COVID, the once easy to collect non-invasive saliva sample is now an infectious medium. But inherent in the virus is a way to protect ourselves, and that is by way of the sensitivity of the viral membrane to detergent. And there are, of course, various guidelines and issuances that will help ensure biosafety. Minimum laboratory measures had to be taken for biosafety, like having that one biohazard room to process saliva samples. From the laboratory to the field and back, we will use a 4-tier sample containment system. The primary tube contains a lysis buffer for immediate cell lysis, all the way to a quaternary packaging system which can ensure waterproofing, shock, or crash proofing. We started using the saliva drop self-sampling method way back in 2007, and the experience then shows that minimum health protocols such as physical distancing can be implemented. Here are photos of partner participants dropping and or checking lysis buffer saliva levels in their tubes. When on-site sample collection is not possible, we can send sample collection kits to partner volunteers. The same 4-tiered packaging system is applied. For sample processing, saliva samples go directly to room 223 and come out as pure DNA extracts. Biosafety measures include protective gear. Pre-COVID, protective gear protected the samples from being contaminated by the DNA that could come from the researcher. With COVID, protective gear protects the researcher from the sample which could contain COVID. The DNA isolation room required biosafety level 2 biosafety cabinets. And after sample processing and DNA extraction, all the minimum laboratory measures for cleaning, decontamination, and sterilization are applied. For sample processing and data analysis, we also had to rely on available DNA samples. Thanks to previous population genetics projects led by Dr. De Ongria, FGRP projects 1 and 2 had 12 EL groups to process. Hence, to date, FGRP1 has genome-wide data for 346 Filipinos, and FGRP2 has 132 whole human Filipino genomes sequenced. And this was possible through the NGS and bioinformatics services of the PGC core facilities. For validation and towards publication, we have the RCEI to facilitate the process. And there is a beyond in the research process because we have biobanking and data banking. We have seen parts of the research process where COVID left a mark. The pandemic has also brought into focus various research ethics issues. the story of Henrietta Lacks and HeLa cells just being one of them. Although for decades, there have always been research ethics issues surrounding genomics research involving indigenous peoples. In the Philippines, we have laws, guidelines, and issuances. However, for the IPRA law and related issuances, it is said that the Philippines is fully compliant in law, but not in fact. Here's hoping that such a research framework would not allow our local genomics research to subject our fellow Filipinos to the kind of genomics research ethics issues that have and are plaguing the foreign research arena. We know and communicate that genomics is failing on diversity, that this can be solved by the inclusion of diverse underrepresented populations. Instead of impressing the indirect and non-immediate benefits of such research, why not just be open, direct, and clearly communicate that we need their help, and that without their help, ancestral diversity will not be represented and genomics will continue to fail on diversity. With that, we circle back to acknowledgements of various institutions and personalities, of past partner participants, and hopefully more partner participants and more partner institutions. And here's to end the talk with a quote from a member of an IP group in Canada. If you're going to work with Indigenous communities on genetics, you have to be willing to make lifelong relations. Thank you for this opportunity to share.
Thank you very much, Sir Fred, for that talk. Indeed, you know, it's very, I really appreciate that your group, your unit has done its work to make sure that even with the COVID pandemic, research, genomics and bioinformatics research as applied to biodiversity, ethnicity, and forensics would still continue, and more so be accessible to us to a wider range of audience as possible. And also, thank you for your work towards making sure that even with the research that being conducted, human dignity and respect for rights of indigenous peoples are also being maintained. So thank you very much for that. Okay, so we now move to our next speaker. Okay, again, reminder, you could ask questions or give comments on our Q&A tab in your Zoom interface. Okay, so I think Sir Fred will be, and even Ma'am Cara will be appreciative of it. Okay, so now for our next speaker, I would like to call to the floor Dr. Ambrosio Din Matias. Dr. Matias, or Din, as we fondly call him, okay, is an assistant professor from the Institute of Biology at the University of the Philippines, Diliman. He received his PhD in evolutionary biology, genetics, from the University of Queensland in Australia back in 2019. His research interest is focused in the use of intraspecific genetic variations in inferring processes that likely influence the distribution of genetic diversity across space. Oh, that's a mouthful. This includes identification of processes, historical and contemporary, that possibly shape the present spatial patterns of genetic diversity in the marine system, and quantification of the contributions of different seascapes in spatial, or should I say spatial, intraspecific genetic variations. Now, to give his talk on the potential challenges in undertaking population genomics research in the Philippines, I now give the floor to Dr. Din Matias. So Din, take it away.
Alrighty, thanks for that one, Sir Jan. Let me just try to, thanks for the introduction, and hopefully I'll be able to break down some of those, some of the things that you mentioned about me. Let me just see how this works. Transition. Oh God, I'm so sorry. There we go. Yeah, hopefully everyone can see my screen.
Yep.
Yeah. Okay. So
Here.
yeah. So again, good afternoon, everyone, and I really I would like to thank PGC, especially you, Sir Jan, for inviting me to this conference. And for this session, I'll be discussing more on potential challenges in undertaking population genomic research in the Philippines. And because I believe that many of us here come from different fields, in identifying these potential challenges, I will try to walk you through the things that population geneticists such as myself, do. So while I work on different systems, really, by and large, my work focuses on marine organisms. So I will be showing examples from marine systems. Okay. So for population geneticists like myself, let me just move this Zoom thingy. So for population geneticists like myself, our focus really is understanding the distribution of alleles across space and time. So for empiricists, what we do is sample individuals from the wild, from their natural population, and identify the state of their genes, that is genotyping them. So the genotyping can involve few genes or few genetic region, or it can be genotyping of the whole genome, which is becoming a norm now, leading to vast amount of genomic data. So this figure here is a simple demonstration of what we can observe when we genotype individuals. So the circle here represent the alleles sampled and from the sampled from the genotype individuals. And by genotyping these individuals, we can estimate the frequencies of these alleles in the different locations, which enable us to make inference about the biological and physical processes which are responsible for the distribution of this allele. For example, how genetically diverse a population can give us insight about genetic drift. How different the frequencies of these two populations can give us clue about gene flow between these populations. Interestingly, genomics is enabling us to examine how these sort of patterns vary across the genome, which help us or which we can leverage to examine selection happening in the wild. So my interest really is understanding how these different physical processes influence these microevolutionary processes and genetic diversity, which to me is an information that can have various applications such as development of a breeding program for aquaculture species to formulation of management strategies for natural population. Using genetic data, however, can be tricky sometimes, especially that physical factors, these factors here, are not constant across space, or sorry, across time. For example, hundreds of thousands of years ago, it is believed that Earth experienced a cycle of glaciation, that is formation of ice glacier, particularly in the high latitude. And this glacier formation can potentially lead to drying up of sea. I don't know why my pointer always disappear. And that drying up of the sea causes exposure of land masses, which at present day are shallow marine area. So for example, this region here are likely became land masses during the glaciation. So the consequence of this glaciation are possible extinction of population in this area, as well as isolation of some regions like this one, isolation of this region because of barriers to migration caused by these land masses. So while these events like glaciation happened in the past, like thousands of years ago, with the last glacial maximum happening around 20,000 years ago, these glacial events are likely to leave imprint to present-day genetic diversity. And it is important really to understand and examine the effects of these historical processes to present-day genetic diversity, not only because they will give us insights into the effects, but also because these potential imprints will affect our inference about other processes such as genetic drift, gene flow, and particularly selection, which is like a big thing at the moment. So for example, in the Great Barrier Reef in Australia, there are efforts to understand the connectivity between populations of marine organisms, such like source and sink dynamics in particular of various marine species for management purposes. So one approach to estimate population connectivity is biophysical model, which integrates biophysical process such as oceanographic current and biological trait to predict where larvae will go. So like all models, however, biophysical model need to be ground-rooted. And one way to do it is to compare its prediction with genetic data. So in the Great Barrier Reef, biophysical model predict a preponderance of north to south dispersal for some coral species like Acropora tenuis and Acropora meliopora. And this preponderance of north to south dispersal in theory can lead to a northern and southern genetic break in the Great Barrier Reef. Unfortunately, in the case of these Acropora species in the GBR, the genetic break or the genetic patterns that are expected from oceanographic currents from that biophysical model are similar to those expected from past events. This northern and southern genetic break that we see might be due to barrier that occurred in this area during the last or during the glaciation. So because of this similarity in possible outcome of historical and contemporary processes, it will be difficult really to attribute the genetic pattern to say biophysical model, or is it because of these past events? And this is where demographic modeling comes in handy, which is one of the tools that I wanted to highlight in doing population genomics. So what we did here is that before comparing the genetic diversity and biophysical model, we first evaluated if the patterns of genetic diversity that we observe is due to historical processes. That is it due to the glaciation, which will lead to this evolutionary outcome? Or is the pattern that we see, is it more due to recent processes, such as the oceanographic current, which can be represented by this stepping stone model? And surprisingly, after weeks of simulation and computation for the two species of Acropora that we work with, we were able to reject this historical divergence model, which paves the way for the interpretation of the genetic diversity in the context of contemporary process or in the context of biophysical model. So I just want to mention that this work is not really based on genomic data, but rather it's based on some microsatellite genetic data from previous publication. And because the Great Barrier Reef, the GBR, and its marine biodiversity is really of high importance to Australia, there were follow-up genomic works for Acropora species in the GBR. And one of our main goals in the genomic dataset for Acropora really is to investigate patterns of gene flow across GBR, as well as poke at potential signals of selection, which again is really important to understand given the rapid changes in our environment. So while examining the genetic data, the genomic dataset for Acropora tenuis, we were struck by a distinct genetic clustering, which is not really surprising for coral. So this figure here show the genetic identity for each individual. So each bar here is one individual, and the color of that bar represents the genetic diversity or genetic identity of that individual sampled from this area in the GBR. So although we can see that most of the yellow cluster individual, cluster 2 as we call them, are from the south, the other clusters here, cluster 1A and cluster 1B, they are relatively widespread, and there are actually individuals that show mix of cluster 2 that are in the northern location. Okay, so regardless, understanding the relationship between these clusters here, this is cluster 1A, cluster 1B, and cluster 2. Understanding the relationship between this cluster, this genetic cluster, is kind of important in making inference about this. I wouldn't want to call it a species, but making inference about this group. Okay, so for example, we're treating this group as one interbreeding species in other ecological studies, such as population connectivity through biophysical model, but they might not be behaving the way we expect them to be. So one potential deviation is that these clusters might not be exchanging genes anymore. That is, there's no more gene flow between this cluster. So one of our initial steps in investigating this genetic cluster is to examine gene flow between them and when they diverge. Again, one nice tool to investigate this cluster is to explicitly model their histories. So what we did here was to examine the population pairwise. That is, compare cluster 1 versus cluster 1E versus cluster 1B, cluster 1B versus cluster 2, and cluster 1A versus cluster 2. And additionally, we examined potential spatial effect by comparing population from the south GBR, from the mid GBR, and from the northern GBR. So the y-axis in these plots here are the different comparisons that population comparisons that we did, and the different plots represent the different parameters for this simplistic model here of population isolation with migration model. So what's really interesting here is that in most comparisons that we did, we obtained a consistent range of divergence time between the clusters. And that divergence time is roughly between 500 to 800,000 years ago, which is roughly the time of the formation of the Great Barrier Reef. Another remarkable result is that the gene flow with cluster 2 is low compared with gene flow with cluster 1. And this is in terms of number of haploid gametes coming into the population. And although this is low, it's still a substantial gene flow. So there's still gene flow between these clusters. There are still a lot of questions that remain to be addressed for this data, such as applying variable gene flow across the genome, or including variable selection across the genome and modeling the history, and so on. But what I really want to emphasize here is the state of genomic research, and where do we stand at the moment, and what can be the potential challenges for us to conduct genomic research in the Philippines. So doing population genomic research involves several steps or processes, and having resources to do these processes, or having resources to do these processes essentially dictates whether we can proceed with this sort of work or not. And one of the initial steps in generation of genomic data, after collection of a sample, of course, will be preparation of DNA library. So this is not generally a problem because it's not that expensive when working with small number of samples. However, if we will work with a lot of sample, which is typically needed for population genetics, then this can be a challenge. So I can't remember exactly how much this process might cost back in UQ. I think it's like 140 Australian dollar, which is roughly 4,000 to 5,000 pesos at the moment. And if you are working with hundreds of samples, say in a corpora that we work with, it involves like 700 sample. The cost of this one will be very substantial. Okay? Once we have this library, the next step, of course, is to sequence them, which is really the costly step here. And this is actually the primary challenge that I see here in the Philippines, because sequencing in the Philippines is relatively expensive. And I know that PGC is doing their very best to keep the cost of sequencing minimal. But if there's no way to lower the price of reagents, then it will be really difficult to keep up with the state of the field, I think. Yeah. And after we generated the sequences, then we can perform initial bioinformatic analysis, such as genome assembly, etc. And from this step, substantial computational resource is required, which I think is another potential challenge that population genomic research might encounter locally. So the assembly of data coupled with some computational intensive analysis will really require good computational resource, that is high amount of RAM, high number of cores, 24/7 power supply, etc. I believe that UP is starting to develop this research through the PGC and, of course, Data Science Common. But this computational infrastructure is something that will really benefit not only empiricists, but also people such as myself who are fond of simulations. Yeah, so while most of the challenges that I see are related to cost, which we can really account for when proposing a project, there are some approaches that as researcher and maybe the policymakers can take so that we can do the same level of research with our international colleague at relatively the same cost. So in terms of molecular work, we can actually do DIY approaches, do-it-yourself approaches, rather than kit-based workflow. And this really proved to be cost effective. So back in UQ, we were able to decrease the price of library preparation, I think, to a third with what DIY approach. And similarly here at UP, I'm starting to implement DIY molecular work as well, which brings down the cost of DNA extraction, for example, to 4 or 5 folds lower. In terms of sequencing, I really do not see an alternative at the moment. My best guess is this is really on the level of policymaker. I think we need to bring the present cost to more than half so that we will be able to generate the same amount of sequences with our international colleagues. Another thing that we can do at the moment regarding sequencing, I think, is to really maximize the sequencing that we do. So prior to performing the sequencing, I think it will be really good to try to plan well how much sequencing effort is needed, like how much coverage do we need per sample, so that we will be able to fill in the sequencing link as much sample as we will be able to. As for the computational needs, I believe PGC and CSRC Data Commons, headed by Dr. John Robb, is taking the lead in filling the gap for this one. So just to give you an idea for the computational requirement for some of the analysis that we do, one of the demographic modeling analysis that I presented required at least 5,600 optimization, some of which can last to 2 or 3 hours, which might translate to more than 20 days of computation if you are working with a 24-core computer. And that is just 1/5 of the typical amount needed. So I just ran out of time in this case, so I just go with that amount at the moment. But in any case, well, it is true that we can ask for funding for computational resources. Moving forward, I believe that there will be a value for a free to access computational resource in the Philippines. As I have mentioned before, there's really a ton of genomic data sitting out there that can be worked on. The microsatellite work that I presented is based on published data, for example. Also, population genomics can benefit from simulation-based work, where computation is the only resource needed. So this might be a bias for me, really, but I would really advocate for this one, especially that most of the bioinformatic and simulation skills that I develop is partly due to me having an access to a massive computational infrastructure that I was able to mess with and actually still messing with at the moment. But yeah, so in any case, these are the main challenges that I see in conducting population research in the Philippines. And again, I do not want to see, or I do not want people to see this as simply a cost problem, but it will be better to think of it as challenges so that we can do a cost-effective population genomic research in the Philippines. And that's all for my talk. And again, I would like to thank PGC and Sir Jan for inviting me to this conference.
Very much, Din. Thank you very much also for accepting our invite for you to give this talk. Okay, and I agree with you. More computational resources for all, right? Right. Cool. Okay. So for information of all, I think Din has to leave in a few minutes, right?
Oh,
right?
Actually, I made a mistake. I thought this was Wednesday.
Oh, so you'll be able to stay. Cool. Cool.
So I'll be able to stay.
So that means, guys, wrap up your questions for Dr. Din, okay, by typing it in the Q&A tab of your Zoom webinar interface, okay? So again, thank you very much, Dr. Din, for that enlightening and very interesting talk, okay? Right now, we're finished with the first half of the proceedings of the day. So we're way ahead of schedule, actually. So instead of a 5-minute break, let's take a 10-minute break for that matter. Okay, so that should give you some time to freshen up, refill in your coffee, get some more snacks, get vaccinated probably. I don't know. Okay, but the main thing is by 2:01 p.m. let's congregate again, and then let's hear the second half of our talk, of our proceedings for today. All right? Okay, so guys, see you in a bit. Alrighty, it says 2:01 now in my clock. Okay, so we now resume our program and get on with the second half of the proceedings for today. Okay, now for our third speaker, okay, I would like to introduce Dr. Lar Ani Morao, Program Director of the Mindanao Satellite Facility of the UP Philippine Genome Center. Dr. Morao received her training at the University of the Philippines Diliman, University of Illinois Urbana-Champaign, Nagasaki University, and Okapi Sciences MD. In 2015, she returned to the Philippines to the Balik PhD program of UP and is now currently a professor at the University of the Philippines Mindanao and as I've mentioned, the program director of the Philippine Genome Center Mindanao. She has led numerous projects related to surveillance of viruses and antibiotic resistance, ranging from wildlife to domestic animals, wastewater systems, and communities. Due to her love for nature, she is also engaged in molecular studies related to wildlife. To present her work on genomic explorations in Mindanao wildlife, I now give the floor to Dr. Lair Morao. Ma'am Lair, the floor is now yours.
Good afternoon. Let me just share my screen. Hold on. Okay, so good afternoon, everyone. Thank you for inviting me to this session on PGC Talks. This afternoon, I will share to you stories on our genomic explorations in Mindanao wildlife. I intentionally titled the talk as Explorations because, disclaimer, I'm not a wildlife biologist. It just happened that I live in this wonderful place in the Philippines, Mindanao, which is very rich in biodiversity. And I happen to be around people who also love to work with wildlife and nature. And I'm also a nature lover, as mentioned in the introduction. So the stories that I will share to you this morning is really the work in collaboration with these people. And I just happen to be around them, putting like a genetic or genomic twist to biodiversity. So our story this afternoon starts with actually a thesis of one of my first students that I handled as a thesis student. So she was looking for a research question, and it just happened that one of her batchmates was also working on bats, and we had samples, potential samples that we could collect, in particular feces from bats. And we were thinking, okay, what else can we do with feces from bats? And interestingly, we found an article from this group in northern Arizona, where they have this very interesting work on species from feces, where where they use feces to barcode different animals. And so we wanted to try this technology or this technique in Philippine bats. Now, why species from feces? This is one possible technique that we can use to do molecular identification of bats. Now, traditionally, we would identify bats using morphometrics, and that could result to some errors, because bats are very small, and so measurements can be very tricky. And in some cases, we would have to resort to looking at their skull features and their dental features to validate their identity. Now, there are some other cool techniques that are around, such as acoustics, which can also help identify bats in the area. And then, of course, we have molecular identification. But with molecular identification, usually we use lethal or invasive methods from, getting tissues from the bats. The advantage of using feces is that it is non-lethal. It is a non-lethal method of getting tissues from the bats. So this technique that they developed uses a very short barcode. It's only 200 base pairs compared to the traditional barcode, which is 500 base pairs. Okay, so we went ahead and sampled bats in UP Mindanao, as well as a nearby barangay, which is in Malagos. And here are my colleagues helping, my bat colleagues helping set up the nets for bat sampling. And this is the bat sampling that we conducted. And in addition to that, so this is also one of the ways where we expanded, awareness and appreciation to students on wildlife by conducting workshops while we were doing the sampling. So we invited students to join us during the sampling and conducted workshops on wildlife, as well as, because my interest is really on infectious diseases. So we also gave them workshops on zoonotic diseases. So can we really use the feces or the poop to identify the bats in the Philippines? And so we sampled the bats, and we PCR using the short barcodes and tried to sequence them. So we were able to amplify 83% of the samples and sequence 62% of the samples. And good news because all of the sequence data showed that the identity of the sequences from the feces really matched the molecular identity of the voucher specimens as well as the morphological identity based on the ID of our bat experts. Okay, so but we did not stop there. We also played around with the sequence data and did some phylogenetic analysis as well as pairwise distance analysis. And one of the interesting things that we saw is that the Asian bats are quite genetically distinct from our Philippine bats, and we see that in both cases. And this was actually also illustrated by our next speaker, Dr. Ian and his group, in also one of their studies. So that was quite interesting because even using the very short barcodes, we can replicate, the information that we can get from the traditional barcodes. All right, so moving on to the next part of our story. By this time, PGC Mindanao has already started. And one of the things that we did during our initial years was to train our staff on library prep and sequencing. So we went to PGC Diliman and had this training, as you can see here. And during this training, we thought, we think we should have like a practical exercise after this training, a really hands-on exercise where we handle real world, real samples and see if we can do sequencing. We can really achieve sequencing in our own laboratory. So while we were thinking of that, we were discussing, okay, which samples would we work on? And then we thought, okay, so we have this initial study on bats, so why not continue working on that? And so we decided to work on bat tissues, and in this case, we used voucher specimens. And we developed the protocol for library prep. So we used the protocol by Jeb in 2015, where they used the strategy of PCR enrichment prior to the library prep. And we sent the samples to PGC Diliman for Illumina MySeq. So this was part of the training exercises that we had for PGC Mindanao. So our wet lab people did this. And once the sequences were available, so it's time for our dry lab people to exercise their what they have learned in bioinformatics. And so they processed the data and assembled them and annotated the genes that were found in the whole genome. And so we were able to get data. And one of the interesting things that we found in the data is about this bat, Cynopterus brachiotis. So this is one of the bats that we sampled in the early study. And this bat has a very interesting story when it comes to taxonomy, because this bat is kind of lost when it comes to taxonomic classification. Cynopterus brachiotis is widely distributed throughout Asia, but scientists are kind of confused as to the taxonomy of this bat, because it appears that in previous studies that this bat has actually many lineages, genetic lineages. And so it's not clear whether this bat is really part of the Cynopterus brachiotis group, or should there be subgroups? Should it be divided into subgroups? So we went to look at the mitogenome data of Cynopterus brachiotis, and this is data that we obtained from our as an output of our training. And so this is the data that we got. We were able to sequence 17,382 base pair of the mitogenome. And when we did the analysis, interestingly, it's the mitogenome is only 90% identical to Southeast Asia Cynopterus brachiotis as well as Cynopterus sphinx. And we did some phylogenetic analysis using the whole mitogenome. Unfortunately, there's not much data available in the database. in NCBI. So we got only a few sequences, but when we compared the sequences, indeed, we confirmed once more, we validated once more that the sequence of the bat we got from Mindanao is really genetically distinct from those the rest of Asia. So that was very interesting. That was a very interesting finding, and we were happy with it because it was just something that we did in our training, but we were able to find something interesting out of that exercise. So now that we have done some exercises on the bat, we decided let's see if we can apply this also to other wildlife. And of course, when we speak of wildlife in the Philippines, one of the one of the very important creatures in our country is of course the Philippine eagle, and it's just right in our backyard. Our neighbor is the Philippine Eagle Foundation. And so we talked to the Philippine Eagle Foundation and proposed to them a collaboration on mitogenome sequencing of their captive eagles, because the Philippine eagles in the those that are bred in the foundation, those are the ones that are used for breeding. And so we don't know at this point, how is the genetic diversity of the eagles that are being bred in the Philippine eagle. But unfortunately, COVID-19 happened, and so we encountered a lot of problems with this project. In particular, very important problem is really the delivery of our supplies and the reagents, because up till now we're still waiting for the reagents. They were supposed to come a few months ago, but there was a problem with the item that was shipped, and so they had to request for another batch of shipment. And things get delayed during this pandemic, especially for us in Mindanao, where shipping gets more delayed. There's a domino effect when it comes to the shipment of supplies. Okay, so that's a very short story that I'd like to share to you this afternoon. But I'd like to close with some of the lessons that I learned from these stories. And the first lesson is that opportunities can come from small packages. So I mentioned earlier, our study just started with a simple thesis study of a student, and then some of the output that we had were really came from our training exercises. And so those kinds of activities we can actually grab them as opportunities to do very interesting types of research. Another thing is that you've seen that there's really not much in the database yet for wildlife. And so genomic databases are really going to be very important in the future. And so we need to populate that all the more, especially for wildlife, and share that data. So open sharing is very important. And that's what we're doing now for SARS-CoV-2. There's global sharing of SARS-CoV-2 sequences. And I think the same should be applied also to wildlife. And that's going to really facilitate biodiversity studies. And then, of course, one of the very challenging things that we are experiencing right now, because all right, we have the facility now in Mindanao, we have PGC Mindanao, our equipment are being upgraded, but then we have the people, we have the expertise, they have the right training, we have the samples, but we don't have the reagents. So there should really be an enabling supply chain so that we can continue to work with our projects, with our initiatives. So yeah, that summarizes what I'd like to share with you this afternoon. And in addition to the people that I worked with, wait, my slide is not working. Okay, I'd just like to thank these institutions and individuals who helped us facilitate these studies. Just very simple studies, but these guys were really very collaborative and very helpful in what we have done so far at PGC Mindanao. So thank you and good afternoon to everyone.
So thank you very much, Ma'am Lair, for that very meaty talk. uh, actually very curious and very interesting na yun nga, nanggaling pardon, I'm speaking the vernaculars, pero sige, I'm more, di ba, parang nagsimula kayo sa, in effect, refuse ng bats, and then, di ba, na-expand. So, it shows that studies in genomes, opportunities come in different packages and even come in the most unexpected ways. And also, yun nga, yung challenge doon sa pag-handle nung mitogenome nung Philippine eagle, sayang medyo na-delay, pero pakiramdam ko na-delay lang naman din ng pandemic, but it will come to fruition kung sakali naman. So with that, thank you very much and also good luck to your future endeavors. Pero sandali, mamaya po hintay po kayo, may open forum po tayo. Okay, so now, so that was Ma'am Lair, Dr. Morao, who gave her talk on genomic explorations in Mindanao wildlife. Okay, now let me introduce the last but definitely not the least speaker. Okay, he is Dr. Ian Kendrick Fontanilla. Okay, so Dr. Fontanilla, okay, who is PGC Diliman's next door neighbor, is a professor of the Institute of Biology under the College of Science of the University of the Philippines, Diliman, and the head of the DNA barcoding laboratory in the same university. He obtained his bachelor's degree, master's degree in biology and genetics in UP Diliman, and his PhD in genetics at the University of Nottingham. He specializes in molecular evolutionary biology, molecular phylogenetics, and wildlife forensics. So related din sa ginagawa ni Ma'am Lar kanina. His lab has been working with the Biodiversity Management Bureau of the Department of Environment and Natural Resources in developing DNA barcodes for Philippine endemic species, as well as handling cases of confiscations of illegally obtained wildlife. He is also involved in the use of eDNA methods to detect the presence of medically important pathogens as well as introduce species in the environment. As a footnote, he is actually the immediate previous program director of the Biodiversity, Ethnicity, and Forensics research program of the UP Philippine Genome Center. So without further ado, to give his talk on the Earth Biogenome Project Philippines, Challenges and Prospects, I now give the floor to Sir Ian. Sir Ian, kayo na po bahala.
Hello, John. Thank you, Sir
Hello.
John. Thank you very much. And thank you for having me here. Let me just share my screen. Yeah. I hope you can see it now. Yeah.
Yes.
So the title of my talk is Whole Genome Sequencing of Philippine Endemic Species, Challenges and Prospects. So I would like to put a disclaimer at the outset. This is more of an appeal. This talk revolves around our appeal, our advocacy towards whole genome sequencing of our endemic species. And I would like to mention why that is, and how hopefully we can move forward. I would like to share the limelight with our project staff at PGC, Biodiversity Forensics and Ethnicity Program, Mr. Enrique Portajada. We made this paper together, this a PowerPoint presentation together. Okay, so let's start with, I may be preaching to the choir, but for those of you who are not overly familiar, I would just like to highlight the sheer biodiversity that we have here in the Philippines. So the Philippines biodiversity is one of the richest in the world. In fact, we are one of the 17 mega diverse hotspots in the entire world. And just to give you a flavor, here for birds, we have 683 known species found in the Philippines, 35% of which are found only here and nowhere else. For reptiles, we have 72% endemicity out of the 365 species. For freshwater species, we have freshwater fishes, we have 24% endemicity. For mammals, excluding humans, we have 240 known species, 58% of which are endemic. And for amphibians, we have 116 species. We have a very high diversity of very high endemicity of 84%. And furthermore, I would like to highlight also the presence of superendemics. Superendemics are those species that are found only in a specific area in the Philippines and nowhere else. So a case in point, we have the tawilis, the Sardinella tawilis, one of two only known freshwater sardines in the world, So this one is found specifically in Taal Lake. And then you have the Isarog shoe rat found, of course, in Mount Isarog. And then the Cebu black shama found in the island of Cebu. And then we could think of also uh, the tamarao found only in Mindanao, in Mindoro, etc. Okay. So just this slide only presents, I'd like to present the sheer biodiversity we have in the Philippines. So we have 13,500 known plant species, out of, probably up to 6,000 are endemic. We have coral species, 421 out of the 577 species in the world are found in the Philippines. So I'm a malacologist. I work primarily on snails. So I would like to share also what are the endemic land snails found in the Philippines. And then of course here you have flowers and several vertebrates. Now, in addition to having a high biodiversity, the Philippines is also a biodiversity hotspot. So biodiversity hotspots are those areas with a great concentration of endemic species, and many of these are endangered or threatened. Why they are threatened? This is because of habitat destruction, primarily because of human activity. So if you look at the red areas here in the Philippines, in the world, this global map, the Philippines, the entire country is a biodiversity hotspot. So we have a very high biodiversity, but we are losing them, okay, to habitat loss. And it is such a great shame that we are already losing some of those that we don't even know are there. So how do we know, how do we measure our true diversity if we don't even know what's out there, okay? So, of course, when we talk biodiversity, we think of the number of species in a particular area. When in fact, in a given area, there are actually three levels of biodiversity. We are familiar with the species diversity, that's the second level, the actual number of species found in a particular area. And then we have the ecosystem diversity. These are the different ecosystems with each with having their own unique set of environmental conditions that cater to specific group of organisms. So if you have a very high ecosystem diversity, you will also most likely have a very high species diversity. But we often tend to ignore the first level, which is the molecular level, and this is genetic diversity. Um, we look at the DNA level, we look at the genomic level, and we can identify or distinguish species just by looking at the molecular level. Of course, this is also in conjunction with other morphological data. So if we are going to evaluate genetic diversity, the ultimate measure of variation and genetic diversity is done at the level of the DNA and the genome. You get the sequence of the DNA or the entire genome. And this brings us to a very important question. Why sequence whole genomes in the first place? Or in other words, why sequence life? So there are three fundamental reasons why we should sequence life. And perhaps some of these may be anthropocentric or human-centered, but it is also connected to our place in the ecosystem and the place of the different species in the ecosystem. So one, we want to revolutionize our understanding of biology and evolution. So we can get very fundamental information from the genomic data. Second, it could help us, the information or the data could help us conserve, protect, and even restore biodiversity. And third, it can also create new benefits for society and of course, human welfare. Okay, and I'll elaborate on that furthermore. So why sequence whole genomes? So we know why we should sequence human genomes. So for disease diagnostics, disease risk, human ancestry, drug design, etc., personal medicine, and forensics, which was already explained by our by Dr. Delphine, Sir Fred, and then Ma'am Cora. But for the perspective of livestock and wildlife, we can use whole genome sequences for disease diagnostics, genome selection, for example, for milk yield and carcass consumption, parentage control, conservation of endangered breeds and species, and disease modeling. For plants, you have genomic selection to improve crop yields, and etc. For insects, gene drives, so that's gene drives, genome editing, and pest control. Fungi, we tend to forget that we derive many, many benefits from fungi. So you have synthetic biology, metabolic engineering for drug production, and other useful chemicals. And then bacteria and viruses. So bacteria for microbiome in health and disease, bioprocessing, detection. And then for viruses, you have vaccine development, gene editing, metagenomic screening, And I think we have realized the significance of whole genome sequencing during the pandemic when we tried to get the sequence of the genomes of SARS-CoV-2 isolates from different patients. So mahalaga ang role nila sa biosurveillance. Okay. So I hope that you are now convinced why we should get whole genome sequences. Okay. Now, what is the current state of whole genome sequencing in the Philippines? So, medyo depauperate ang ating data, ang ating database for the Philippines, despite the fact that we are a mega diverse country. At least from the publications or initial survey that we conducted, whole genomes have been done or conducted for the Katigan dwarf of Cocos nocifera, which is the coconut. This was done by our colleagues from the Philippine Genome Center. And then Musa textilis or abaca, and these were just recently published. And then majority of whole genomes concentrate on mitochondrial genomes for the simple reason that, well, they're easier to obtain because they are smaller for animals, 15 to 17,000 base pairs. And of course, it's cheaper. And then it's informative enough to derive evolutionary history. So this is what we have obtained so far. So we have Tarsius silicta, the risota, the land snail, Cynoptus brachiotis. I think this was mentioned by Dr. Lyer in her talk. The Philippine, the crab-eating macaque, and then the Rosetus amplexicaudatus. So there are also efforts to conduct the whole genome sequences of plastid DNA. And I am aware that our colleagues from UST have already done that. We're just waiting for the publications. But there are sporadic efforts across the Philippines to do mitogenome and plastid genome sequencing, but there are far fewer efforts, if any, for whole genome sequencing, at least from the perspective of eukaryotic organisms, multicellular eukaryotic organisms. There are already people working on whole genome sequencing of bacteria and viruses. Particularly the Philippine Genome Center, they're working on the viral genomes of the SARS-CoV-2. So this is where the Earth Biogenome Project comes into play. This one is an international consortium, or the EBP, which is a moonshot. If you're familiar with the term, an attempt to go to the moon for biology that aims to sequence, catalog, and characterize the genomes of all the Earth's eukaryotic biodiversity over a period of 10 years. So they have already started in 2018, and then it's already 2022 next year. So that's roughly a little more than 6 years left, okay, in the timeline. But I suppose they will extend that beyond 2028. Now, its aim is to produce an open DNA database of genomic sequences that will provide a platform for scientific research and biodiversity conservation efforts. So, maraming kasama dito, institutions and even countries. But the Philippines, as of yet, is not a part. But we are in the process of becoming a participant in this global endeavor. So what are the project goals and anticipated outcomes of the Earth Biogenome Project? Well, the first one is to revise and reinvigorate our understanding of biology, ecosystems, and evolution by utilizing the whole genome data. So what are its implications? It will give us a better understanding of evolutionary relationships among all known organisms, allow us to fully elucidate the timing, origin, distribution, and density of species on Earth, generate new knowledge, ecosystem composition functions. So these are basic research, okay, which will hopefully become a springboard for applied research, okay. And then number two, enable the conservation. Ito na papasok yung application. So enable the conservation, protection, and regeneration of our biodiversity. So particularly when in the context of climate change, and then clarify how human activities and invasive species affect our biodiversity, develop evidence-based conservation plans for rare and endangered species, and create genomic resources to restore damaged or depleted ecosystems. And thirdly is to maximize returns to society and human welfare. So of course, many of the whole genome projects across the globe are nationally funded, meaning they are funded by the government, and therefore there should be some direct applications to society. hindi lang pwede na basic research lang. Of course, basic research is important, but there must also be a what you call return of investment or ROI. So how does that work? Well, as I've mentioned, we can use that to discover new medicinal resource for human health, enhance control pandemics, which we are now witnessing. Okay, because of genetic information, whole genome data, we are able to conduct biosurveillance and, more importantly, characterize different varieties or variants of these of the COVID-19 SARS-CoV-2 virus, and whether it can be more effective, whether it can be transmitted more easily, or it will evade vaccines. And we know that based on the activity of the viruses vis-a-vis its genome. And then we can also identify new genetic variation for improving agriculture, discover novel biomaterials, new energy source, and biochemical resources, and improve environmental quality. Okay. Now, the EBP has 48 affiliated project networks around the globe. So the scope of each project is very comprehensive. It's usually geographically centered. For example, you have in Chile, they have the 1,000 Chilean Genomes Project, meaning they aim to whole genome sequence 1,000 samples or 1,000 species. And then you have the 1,000 fungal genomes, the 5,000 insect genomes, the 10,000 bird genomes, the 10,000 plant genomes, and the Africa Biogenome Project. So they are now talking about in the number of thousands already. Whereas here in the Philippines, we haven't even started a full-blown whole genome sequencing project aside from what we did for the coconut and the abaca. Okay, I think I may have. I'm not sure if I've missed any other whole genome projects, but there is no concerted effort to whole genome sequence our endemic species, which is also a legitimate resource of any country. Now it has the EBP has 45 institution members around the globe. We hope that the Philippines, through the Philippine Genome Center, can be a part of this network. So as one of the 17 mega biodiversity hotspots in the world, the Philippines should be at the forefront of whole genome sequencing of endemic species. So this is the organization model of the EBP. At the very center is the coordinating council with experts around the globe from UC Davis and from Australia and in Canada. So, and then around here, you have these different projects with different focus, target taxa, and the size or the number of target taxa. And then these are the regional networks. So you have European Union, Australia, China, Japan, Canada, Africa, USA, and Brazil. So if you notice, it appears that Asia is underrepresented because it's only represented by Japan and China. There's no representative from Southeast Asia, which again, I'd like to highlight as a very high biodiversity. And many areas of which are biodiversity hotspots. So if we are going to embark on genomics biodiversity research, there are several steps by which we will have to go through. Of course, the very first step, which is the most difficult step, and I can speak for every scientist here in the audience, to draft a research proposal, writing to gather the momentum and conduct an actual research. You have to come up with an approved research proposal, and writing a research proposal is just like writing a PhD proposal or an MS proposal, and then I'd like to highlight, however, that here in the Philippines, we have this misconception that funding is scarce. In fact, we have enough funding. The Department of Science and Technology seems to be getting sufficient amount of funds. It's just that we don't have enough warm bodies to write the proposals and conduct the research. So we really need to generate more scientists to conduct our research, and we need them to write these proposals. Because otherwise, if there is no snowball effect in terms of the need to come up with a bigger budget because there's not enough warm bodies, then the government will not be compelled to increase the budget for research if there's not enough justification, if there's not enough demand to increase the research. Okay. Now, the second step, secure permits. This is also very important because we have to respect our biodiversity because this is a national patrimony. It's not an ownership. It's not owned by single individuals or groups. Well, except for the IPs, they have primacy over the biodiversity found in their local areas. But as a whole, biodiversity is owned by the Philippines as a whole. By the Philippines, so and its people collectively. So therefore, we have to follow the necessary process of securing permits para hindi tayo magkakaproblema sa pag-publish, and most importantly because they contain uh, information that can be economically important, especially from the perspective of bioprospecting. So dapat we have to have the first take as a Philippine, as a people on the resources of biodiversity derived from biodiversity. And then step three, there's human resource development. This is very important. In order for us to have enough research output, we have to have enough people working on research, and therefore we have to produce enough PhDs and MS students. So we need to come up with more programs, specifically in bioinformatics, to analyze this volumes upon volumes of data that we will eventually generate. And then yung step four, I think once we start embarking on collective efforts towards whole genome sequencing of our endemic species, we have to also think of or consider biobank and biorepository. Where do we store our specimens? There are protocols by which endemic species or those that are sequenced are properly annotated and identified where they are stored, particularly with the proper accessions in the museum. So we have the UPLB Museum of Natural History and also the National Museum. We should deposit our biological specimens there, and of course our genomic resources, yung ating mga DNA. We should also come up with a biorepository or biobank for such materials. So in step five, we nucleic acid techniques, DNA sequencing, and bioinformatics analysis. Of course, these are standard protocols, and I won't belabor the point, provided we have the money, of course, once we have the funding. And then, of course, publications and returning to the local communities, especially where we derive our taxa or our organisms or our resource materials, we should always go back to the community and feedback, provide feedback, and more importantly, they should also benefit. Okay. Now, I'll just give you some example of cost estimation of how much really does it cost to whole genome sequence an entire species or just one sample. I would like to take note that this is just a rough estimate and the prices are subject to change. Now, I have given here some examples, yung Philippine Eagle, which I hope the PGC Mindanao and UP Mindanao, with the help of the Philippine Eagle Foundation, will push for this, the whole genome sequencing of the Philippine Eagle, because the Philippine Eagle is our national bird and is critically endangered. Now, just to give you some perspective, its genome size around 1.28 gigabase pairs. Now, if you're going to do roughly 30x coverage, which is reasonable enough to produce a whole genome sequence data, and if you're going to use a standard, the NovaSeq SP300 sequencing kit, and let's say we have six samples to run in order to get the whole genome sequence of one individual, it will cost around 433,192 pesos. Not bad. But of course, that is just for this sequencing kit. It could go up to 2 million depending on the sequencing kit and the quality of the data. Now, if you're going to look at the Philippine O3, which is roughly the same genomic size as the Piticophaga jefferi, ganun din ang presyo, 433,000. Now, the Tarsius or Carlos Carlitos Siricta, which has more than twice the genome size, you have 30x. We can reduce the number of samples to bring the level down to 397,000. But of course, if we increase the number of samples, we will provide we will definitely have greater resolution, and of course, it will also cost us. So these are just to give you some idea. This is just the sequencing part. Hindi pa dito kasama yung bioinformatics. Of course, bioinformatics analysis will, and the annotation of the whole genome data will also take some time and manpower. So kailangan po nating i-develop yung ating manpower in order to analyze this data. So from the perspective of capacity human resource development, we already have the Philippine Genome Center, and we see the relevance of having a Philippine Genome Center, especially in the context of the pandemic. But hopefully, I hope you will also realize the significance of having a Philippine Genome Center when we look at biodiversity and study our biodiversity. Now, the PGC has already expanded. We have originally the main headquarters here in UP Diliman, where majority of the facilities are present, particularly the DNA sequencing core facility and the core facility for bioinformatics. And then we have now expanded to UP Visayas and UP Mindanao with their own core facilities in the hope that we will bring Philippine Genome Center closer to the Filipinos, wherever they may be. And then meron na rin pong expansion plan na nagaganap sa UP Los Baños, which will focus on agriculture. So in terms of human development, human resource development, there are programs in the work, the MS and PhD bioinformatics programs, which are proposed to be offered across the different campus units of the UP system. So meron po sa UP Manila, UP Diliman, UP Visayas, UP Mindanao. So you don't have to go all the way here to UP Diliman. And because of the nature of work in bioinformatics, in most cases, this can be done remotely. Baka you don't even have to leave your own area or your residence just to conduct your MS and PhD bioinformatics program. So the target implementation for UP Diliman is next school year, 2022-2023, because it's now the proposal already in the curricular cluster committee. So it is our hope that it will be offered next school year. And more importantly, I invite those who are out there in the audience to enroll in this program if you are interested. So just to give you some perspective, the MS Bioinformatics is 36 units, and for full-time students, it's supposed to be 2 years. I say it's supposed to be, kasi alam niyo naman yung thesis tsaka dissertation, it could be from 2 years to forever. Okay, so para ma-level the expectation nila. And then PhD Bioinformatics, for those with an MS degree, it's only 37 units and 3 years. And then 61 units and 4 years for those with the BS degree only. So you can have a straight PhD bioinformatics. Okay, so to wrap up, so the Philippines is one of the major centers of biodiversity, can participate in the whole genome sequencing efforts. So we have already conducted a series of meetings with potential collaborators within the University of the Philippines system, and we also want to expand this to other institutions and government institutions, state universities and colleges, and higher educational institutions, because this is a national effort, and it's not a sole burden of a single institution. So we have already initiated the Philippine network of Earth Biogenome Project. We are not yet a member of the Earth Biogenome Project, but we are already moving forward to have initial projects so that we can qualify as participants or members of the Earth Biogenome Project. And then we will secure funds through the DOST, CHED, DA, and if there are international funding agents, funding available, then we will also do that. So it is our hope that those in the audience will participate in this effort, either as research assistants or collaborators, or as our MS or PhD students. Okay, because there's enough for everyone. Because of the sheer biodiversity of the Philippines. We will never finish our efforts to study the genomic sequence data of Philippine biodiversity in just one generation. So to summarize, so dito ko nasasabihin yung challenges and prospects. So of course, there's limited data on Philippine endemic species. I'd like to emphasize that funding is not a problem, but we need people, warm bodies, to write and apply those grants. Because if the government does not see the need to increase funds, meaning there's not enough people writing research proposals, then they will not be compelled to increase research funding. There's also a need to effectively communicate the value of whole genome sequencing, and I hope I have already convinced you why we need to obtain the whole genome sequence data of our endemic species. And then, of course, we must also observe ethical practice, collecting permits, and especially in areas where there are indigenous peoples, we have to go through the correct process. And I must emphasize correct process and correct institutions, not dapat lapitan. And there's also a need to utilize museums and biobanks for storage of biological material. So we have to systematize how we need to collect and store our resources, both molecular and biomaterial resources. Now, in terms of prospects, there's a huge opportunity to work on a diverse group of taxa. There's not enough warm bodies to cover all the biodiversity. So now is the perfect time to start. In fact, it's already late. Dapat nag-start na tayo a generation ago pa. But nevertheless, now is the perfect time to start. And then we hope to form collaborative networks within the Philippines and outside through the Earth Biogenome Project. And there is opportunity for human resource development in the field of genetics, genomics, and beyond. So with that, I would like to thank you for listening to our talk, and please join us in this national and global effort. Maraming salamat.
Maraming salamat rin, Sir Ian, for giving that very interesting talk. You're right. Philippines being a biodiversity hotspot, okay, and a recognized biodiversity center globally, okay, it is actually vital that we also take advantage of, um, you know, of of that ano ba tawag dito, richness, no, of species of the biotic sphere, biosphere, right? To be able to generate knowledge that will eventually help, you know, the Filipino people and even those abroad understand the richness and the diversity of yung ano natin, environment natin, as well as yung mga flora and fauna here in the country. Also, hopefully the Earth Biogenomes Project could help bridge that gap. Also, thank you very much for advertising the MS and PhD Bioinformatics program that we've been cooking for more than a decade now. I'm glad that it has already taken off, at least preliminarily. Hopefully, we could get it off the ground and offer it by next school year. Okay, and also to our audience, there you go. There's a call to join in and to study, right? By the rich biodiversity of the Philippines, and hopefully some of you would be able to heed that call. So thank you very much for that, Sir Ian. Okay, and now that takes care of our the talks that were of our different speakers. Okay, now I know there's an open forum coming up, but before that, I would like to share with you something that the program committee of the Genomics Bioinformatics Conference has prepared for quite some time now, in the hopes of contributing towards overcoming the challenges and building opportunities towards enriching the human resource aspect of genomics and bioinformatics. And this is the GB Learn web application. Okay, so you have there the link or the URL. Okay, it's gxb-learn.pgc.up.edu.ph. Okay, it's actually a web based application, but you could use your mobile devices to view it. Okay, and it is meant as a learning application for genomics and bioinformatics, right? And it contains, at least it will eventually contain various tabs on genomics and bioinformatics. Okay, the content of which are provided by the PGC's very own DNA sequencing core facility team, as well as the core facility for bioinformatics team members. Okay? So actually, I think the link is now on, although there's still the content is still scarce at this point in time because we're still going with the editorial process of making sure that the content is of top quality, and so as to help budding scientists and even senior researchers learn at least the fundamentals of genomics and bioinformatics, right? And the target audience is from probably from senior high school up until very senior researchers. All right. So again, it's gxb-learn.pgc.up.edu.ph. Please do check it out, and we're excited to share this with you in hopes of that the genomics and bioinformatics could be promoted and knowledge on these fields could be facilitated. All right. All right. So with that, okay, with that, so that ends the plug. We now go to the somewhat interactive portion of our proceedings for today, the open forum for questions and answers, right? So if you checking the Q&A tab here in the Zoom interface, it seems that a lot of people have been shy in posting questions, or at least they haven't gotten around, developing or crafting their own comments and questions. But our PGC team, the program committee team members, have been also busy soliciting questions through our back channels, right? And we will be asking them, at least some of them, to the speakers for today. So with that, I would like to invite Ma'am Cora, Sir Fred, Sir Din, Ma'am Lair, and Sir Ian to kindly open your video cameras so that people can see the lovely faces of our speakers for today, right? And also, more exposure. Kumbaga, baka ma-discover. Malay nyo, may talent scout dyan sa mga audience. All right. Sige. So, let's see. Okay. So, yeah. So, pakibigyan na lang po ng spotlight si Ma'am Cora at saka si Sir Ian. Yeah. Okay. Sige. All right. So, for the audience members, if you still have follow-up questions or other questions, feel free to chime in dun sa Q&A tab. So, I'm going to be reading yung mga back channel questions. Okay. So, first, this is for all speakers. Okay. Okay. This is a three-part question. So first, are there many people in the Philippines with your expertise in the sort of bioinformatics analysis needed to analyze genetic and or genomic diversity data? How does someone interested in that type of research learn how to do that? What sort of prerequisites would people need to already know? And are you aware of any resources people can avail of to learn more? Okay, so to start off, siguro sa screen ko, yung pinakamalapit sa akin, si Ma'am Lair. So Ma'am Lair, kayo muna pa.
Actually, that's a very good question. And this is a good opportunity to call on the young ones to explore the field of bioinformatics, because we do lack experts in bioinformatics. So in our experience here in PGC Mindanao, when we look for people to help us in bioinformatics, only a few respond to the call. So we do need more young ones who will venture into bioinformatics. As to what are the requirements, that question was asked to one of our bioinformaticians, and I'm just going to share what he said. And he said that it's more of a good background in computational science, and maybe some background in biology, but I think what's more important is a good background in computational science.
Right. Thank you very much, Ma'am Lair. Sir Fred, how about you? Ano po yung masasabi niyo?
Yeah, there were three questions, right? I guess totally agree what Dr. Morao said. It would be better really to have a background in computational science. You see, what we do in genetics, they're pretty much theory already, they're laws and what have you. But the adaptation of a biologist to computational versus the adaptation of the computational scientist to biology is faster. And apart from having a good background in computational biology, computer science, even logic, what have you, it's actual practice. So if you want to learn and you want to contribute, yes, join in the research, because just like any other workshop that you attended before from Perl, C++, if you don't get to practice that, then it'll get lost. So it's best to participate and find your niche. You want to work on humans, you want to work on animals, and what have you, then yeah, participate, join the research group.
All right, thank you, Sir Fred. So you heard that. Get your hands dirty, guys. Get your hands dirty. Okay, Sir Din, how
Yeah.
about you?
Yeah, so I totally agree with what Sir Fred mentioned. It's really about continuously working on your skill, continuously applying your skill. But I just want to share my experience because I started without knowledge in bioinformatics. When I started my PhD last 2015, and I came into a lab that is just digging or just starting with genomics as well. So we were like lab rats basically. My friends and I, we are lab rats in that lab. So no one in us we don't know how to process like next gen sequence and everything. So essentially, we don't know anything. And I think one big factor really is motivation and perseverance. I think that's the thing that I would I think that's the game changer for all of us. The resources, they're out there. You have Stack Exchange, whatever forum that you can use to ask questions. There are very responsive people. I used to be one of the top 2 or 1% in responding in Stack Exchange when I have time. but resources is out there online, so it's just really about motivation to do work. Yeah, and once you the learning curve is quite steep, but once you get there, like what Sir G mentioned, you really need to continuously practice it. But regarding the background, I think I believe everyone can learn it. We just need to find our motivation to kind of learn it. Like what I've mentioned, I started from zero knowledge, and until now, I'm one of the go-to when it comes to demographic modeling for population genetics. And yeah, it's all about like self-learning. And in my case, it's like self-learning, asking questions, and continuously working on things. Yeah.
All right, thank you very much, Dr. Din. So guys, motivate yourself. And I hope this the 3-day conference would help you would have inspired you to, take up a career or at the very least help out in these kinds of fields, right? Okay. Ma'am Cora, kaya naman po.
Okay, thank you, Dr. Yap. I think I'm the old school. Kami iniiyan that we need to go back, review. Always reminding ourselves, lifelong learning. Dapat graduate na ako dyan, pero hindi ako makagraduate dan. Kailangan balik. And that's something to always remind ourselves, regardless of which part of the hierarchy of learning we are. We need to always go back. It's lifelong. And also to have that humility that there could be interaction between the young and the not so young in order for both parties to learn. It's not just the young learning from the old, but also the old learning from the young. And I think that's one thing we need to establish at the university and also amongst all the different interested parties is the 3 C's: cooperation, collaboration, and communication. Because we can learn from each other in terms of maybe somebody is very good with modeling, ibato mo na lang sa kanya. Or maybe we need to have that support. And sometimes we forget because we're so busy, we forget to look out for each other. And that's a struggle. So that's part of the learning. And then to build that community of genomics lovers, that we love genomics, but to always remember the purpose of why we do things. If we're so immersed with trying to do the model, trying to sequence, pero always ask yourself, bakit ba? Why am I doing this? And never to lose that focus that really what we're trying to do, we're trying to generate knowledge, not to be able to graduate alone, not to publish the paper alone, but in order to make life better for the rest of us here and the next generation. So that we need to be convinced, kahit mahirap yan, late nights, every day, numbers, tubes, data, etc. But do not forget that it has a purpose. I think that's my main thing. Thank you.
Thank you very much, Ma'am Cora. I agree with you, diba? Apart from how to do it, para in a sense, why are you doing it or for whom are you doing it, diba? And it should be talaga for the service of, the larger populace talaga. And also, I agree with yung collaboration, diba? I mean, one of the keywords sa sa themes natin is diversity, diba? Biodiversity. Bioinformatics and even genomics itself is multidisciplinary and parang it's a diverse set of knowledge areas talaga, and it's important na merong collaboration, merong interaction talaga, and that yun nga, lifelong learning has to be an integral part of what we do. So thank you very much for that, Ma'am Cora. So Sir Ian, kayo naman po.
Nanalo na sa Q&A portion ng Miss Universe. Thank you, Ma'am Cora. Anyway, ako naman, I'm an evolutionary biologist, and I'm a biologist by training. So I'm not a bioinformatician. So I really have to work with bioinformaticians in order for me to find out what data is available there, and hopefully be able to understand these biological processes, evolutionary processes, and how we can benefit from them. So ultimately, kailangan talaga natin ng bioinformaticians dito sa Pilipinas, and it is our hope that it is easier for the next generation to learn bioinformatics, unlike tulad natin na mga not so young but not so old yet, who have to go abroad to study bioinformatics. So I hope that there will be a day that because of the sheer biodiversity we have here, we have more than enough material to work on, and there's already infrastructure, and both physical and your academic wise, that will provide avenue for our students to pursue bioinformatics. So meron na po tayong data QP, data commons, para magamit yung mga servers, of course for a fee, and then we also have these graduate programs all over the UP system. Of course, ina-advertise ko yung nasa UP because I'm from UP, so that our audience will know what's available. So you can pursue bioinformatics whilst you're here in the Philippines. So sana mahikayat kayo. We already have, nagkakaroon tayo ng snowball effect. Bumabalik na yung mga nag-aaral ng bioinformatics abroad, and they will be the next generation of experts. So nandito yung mga kasama natin, bioinformaticians, especially Dr. Din Matias. He was my undergraduate student, would you believe? And we were strictly biological, yung aming ginawa. And I'm quite happy with the trajectory of his career development. And he's a very, very welcome human resource in our institution. So I hope we will have more of Dr. Matias from the audience very soon. Thank you.
Okay. Thank you very much, Sir Ian. So yun nga, in a sense, yun ang importante na magkaroon tayo ng local, diba, mga institutions talaga na that would help facilitate, diba? Nandiyan na rin naman yung resources, both equipment. Siguro may kulang pa, pwede pa naman madagdagan, diba? But in terms of knowledge, na sabi nga ni Din kanina, diba? For example, na the information age because of yung sa internet, diba? Industry 4.0, internet, yung availability ng information at at your fingertips, that also help talaga entice or build at least informally yung knowledge capacity ng local scene natin. Okay, so thank you very much, Sir Ian, for that. Okay, so we have another question sa back channel muna. Okay, this one is for Drs. Matias, Morao, and Fontanilla. Okay, so the question goes, we've seen a lot these past 2 years about how the pandemic has affected the health and behavior of human populations. Have you ever investigated or are you aware of any studies on how the pandemic has affected wildlife populations? Okay, so sige, si Din muna. Unahin ko this time.
Okay, yeah. I haven't really delved into this topic, but I know that there are people who examine recoveries of ecosystems during pandemic because, for instance, there are less basically humans were kind of restricted to go out, go to nature, and so on. So there was some sort of recovery time for natural ecosystem, but I haven't really go into those works, but I know that there are people working on it, and I think there are some publication about it. And I can't say much about what they found out because 2 years is kind of like a short time for recovery, I guess. Yeah. So that's all I can say. Sorry.
No problem, Din. Thank you very much for that input. Ma'am Lair, how about you po?
Yeah, same with Dr. Ding. I don't think there's any report that I've seen so far on the effect of the pandemic on wildlife or on nature. But it is a very good research question. That's something that we can explore at this point. I just recall at the beginning of the pandemic, there were posts here in Davao, ha. I don't know in other parts of the country, but there were posts about sites of animals, in urban areas, makita lang nasa daan na ganon. So, baka baka habang yung mga tao sa bahay ay nagpa-party na yung wildlife natin. So that's really something interesting. So I don't know, we haven't confirmed that, we haven't validated that. So again, as I said, that's a very interesting research question. Pero the other story naman, the other way around is studying how wildlife can affect pandemics. We do have studies on that, looking at zoonotic diseases, potential emergence of these diseases from our wildlife. So we have ongoing studies on that right now.
Thank you very much, Ma'am Lair, for that input. So Ian, kayo naman po.
Very similar lang din yung nalalaman ko kay Ma'am Lar at Doc Tin. Pero I do know that there was at least one study done in China that conducted the dispersal patterns of the Oncomelania, which is a opensis, which is a vector host of schistosoma. So they saw a difference in the dispersal pattern pre-pandemic and post-pandemic. And this has something to do also with the restricted movement of people in urban areas, in urban areas where there are waterways. So yeah, yun ang may personal knowledge ako. Pero hindi pa na yung just wait for the publication. I'm not part of the study, by the way, just a disclaimer. So but like yung sinabi ni Doc Lair, I think maganda yung tingnan natin yung effect ng wildlife, particularly with the encroachment of human habitat with those of the habitat of wildlife, which increases the chance of dispersal of new diseases or novel diseases. So at least yun yung pwede nating pag-aralan.
Can I add something?
Yes, go ahead.
I think this kind of question, citizen science can contribute to this really. And I think if any one of you try to download the app iNaturalist, it's a simple app where you can use your phone to document biodiversity when you're walking around your house and so on. So those data, they go to a common database, a public database like GBIF. And we can answer these questions if we just pull out data from GBIF. And this is one of the things that I really want to advocate, because temporal data sampled from the same locations will be really helpful to address various ecological and evolutionary questions. So it's actually one of the things that I want to start with high school. Like across the Philippines, it's something that can be done, incorporated in their activities, which will give us continuous temporal data. We can monitor changes in our ecosystem across time. So if you have that system prior to the pandemic, addressing this question, that data that could have been gathered during the pandemic could have helped in addressing this question. So if you are fan of walking, going to the nature, I would suggest download iNaturalist and start documenting what like the critters that you see up there. Yeah, alrighty. That's all. Thanks, Sir Jan.
Thank you very much, Dr. Dean. So citizen science, that's very interesting, but that's basically engaging, um, basically, making inclusive, making
Yeah.
genomics, wildlife science inclusive, biodiversity inclusive. Just so ano rin, meron po ba kayong input, Ma'am Cora and Sir Fred, dun sa ano? So, Ma'am Cora, may input po ba kayo dun sa
Actually,
question?
I'm interested kay Dr. Liar because you study bats. Did you have anything, I don't know, anything on bats in COVID? Or was there any screening of animals that could potentially be another animal host of the virus?
Yeah, so we had actually prior to the pandemic, no, we had studies in bat looking for coronaviruses here in Davao, and we did detect coronavirus like viruses in bats. But we still need to generate like whole genome sequence of these viruses, so we can really understand them more. So that's what we're trying to do right now.
Very much. So question, at least ano, may question within the speaker's pool. Sir Fred, meron po kayong dadagdag pa sakali?
Actually, in the first year of the pandemic, there were reports of zoo animals getting COVID, like tigers, cats even. More recently, there's actually a news that even hippos have acquired COVID. In Mongolia, I think there's this, I forget the type of animal, but squirrel type, rat type animal that's endemic to Mongolia have also been reported. But it was highlighted because it was part of the diet of the people in that region. So they issued a warning that, okay, stop eating this kind of animal first, unless because COVID was identified in those animals. But how they acquired it, yeah, that's the question. Zoo animals, would they have acquired it from humans? I don't know.
Thank you very much, Sir Fred, for that for that input and additional tidbit right there. Okay, right. So we move to the next question. Okay, this one is specifically addressed to Sir Fred and Ma'am Cora again. Okay. Again, it's a three-part question. Okay. In your work on genetic and genomic studies of human populations, what sort of obstacles do you encounter with regard to education, outreach, and receiving consent? Do you find that people are generally interested in participating and willing to provide samples? Does it often present a problem when people are unfamiliar with basic concepts in genetics or molecular biology? So, Sir Fred, would you like to start?
I think the
No?
answer to all those questions is yes.
Yes.
Yeah, yeah, it is difficult working with humans. Take note, with, not on. We're trying to change the philosophy, meaning working with them would actually suggest them participating in the study, especially with indigenous peoples and vulnerable groups. Education, I've often said more recently that the consent that they provide is not really because it's not really because they understand what you've communicated at the start or during the social preparation stage. Because if it took us here in the panel years to understand our craft, we should not expect that during the social preparation and the free and prior informed consent process that they will be able to digest all the information out of full disclosure that we give to them. So pretty much their consent is really based on trust. You can put that at like 99%. And it's how you conduct the research, how you treat them, how do you not abuse whatever research results you have, and following the entire process appropriately and dutifully. Yeah, it's quite difficult. We've encountered yes and no's in terms of participation. We will not expect that they will be able to digest every information that we give to them. That's why the philosophy is rather changing right now. It's really building relationships, and not just okay, you oriented them, then you go back to your lab, you do your DNA, and then you appear later on. Sometimes other research groups don't even come back and report results. In the reporting of results, especially for our Filipino population and ICC, indigenous peoples is not just reporting results. You have to discuss the results with them and they have to approve it. If they don't approve, you don't have any publication. So it's not just going back and reporting, you discuss and they have to approve it. So to some extent, they become co-authors in the project. So it's really a change of format of your relationship from the start to the finish of the research process that you have to account for. Not just, I think Dr. De Ungria mentioned it eloquently a while ago, it's really thinking about the Filipino people in our case, and the problems that I mentioned in my talk earlier about what is encountered in the foreign arena. This is something that we'd like to already get to the attention of the Filipino people so that they will be aware of these problems, and to some extent you give them full disclosure, and therefore their consent would be more relatively genuine. So yeah, to those three questions, yes. The answer is yes to all of them. And as I've explained, the different factors that we have to consider in doing research with human participants.
Thank you very much, Sir Fred. Interesting that you mentioned that they also become, contributors or co-authors. So instead of them just being the subject, they also are an active participant in the research process. That's an important aspect because they are the source of the data after all, and they could to be able to contextualize data, no better way than to ask the source themselves, in a sense. Yes. Okay, so Ma'am Cora, any additional input?
Actually, more than just participants, they are the owners. We need to recognize that they own the data. Therefore, their consent is needed. Without that, you cannot use their data. So we're just, and it's important for us to recognize as geneticists, as genomicists, whatever, we are only steward of their data. Whether this is IP or not IP, it's not ours. So this is a different perspective in terms of research. When you say, this is my sample I collected, I got the funding, it is my sample. I move to another institution, it is my sample, I take it away with me. When I go to biobank, I will determine who I will share it with because it is mine. No, that is wrong. So it's really the recognizing that they are data owners, and we are only stewards. Also, in the point that Fred made in terms of trust, it is true, the free and prior informed consent of many people, pati nga tayo, hindi rin natin naiintindihan lahat eh. Kaya tayo, sige, payag na ako, wala naman ako naiintindihan. Payag na ako, hindi ko man naiintindihan yan. You know, it really, and particularly with sophistication of data analysis, computational analysis, even people at the university could not fully understand. So what more the ones who have not been able to experience as much as others have experienced. And one thing that I have in that trust, and that's where communication comes in. One thing that we need to teach our science students is how to communicate. Communicate not only in jargons, but also communicate from the different levels of the hierarchy. From the policymakers to your peers, to your faculty, to your researchers, down to the people, well, not down, across the communities. There are times when we find ourselves, when we do forensics, we do population genetics, you're talking to the president of the university, you're talking to the member at the European Union, 1 hour later, you're in the prison talking to the people in Bilibid. And you need to be able to adjust the language. And one skill that I advise for the younger ones is that we need to talk from the language of the heart. Because we may be translating all your RCEIs in Ilocano, Tagalog, etc. We may be speaking English, in whatever language, but we do not put our heart there, then we are not communicating. Because there is universality in what we aspire for. Whether we are IP or not IP, whether we are researchers or not researchers, we aspire for the good of ourselves, of our families, of our community. That's what we need to touch. And that one thing for the ones who will choose, I've been in this profession for the last 25 years. So I will be passing the baton very soon. But for the young ones, I think one thing that we need to really appreciate is this type of work with research, with with genetics, with IPs, etc. As we said, it's lifelong learning, but it's also a lifelong commitment. It is not that they learn from us. We learn from each other. And I hope that those ones who are going to the field will realize you want to give. No, actually, you're taking much more than what we're giving because you learn much more. And I think in the pandemic, we realized that the value of family, the value of persons, the value of integrity. I think those things we need to recognize when we choose the research that we would like to undertake. Thank you.
Thank you very much, Ma'am Cora. Actually, napaka no? Parang may lalim na pinaghuhugutan. two things though. Ako kasi I also work as quasi data protection officer. So tama yung sinabi niyo po na the sources of data are the data owners and we are just data stewards. We have to respect na hindi tayo ang may-ari. Secondly, yung importance of communication. I guess parang meron kaming joke sa or some sort of parang funny anecdote palagi sa computer science or programming na we don't do documentation. Yung parang bad practice noon, I would like to think noon yun na bago na ngayon. When we do code, we don't document and we don't have to explain yung tag dito, yung functions, because it was hard enough to program, it should be hard enough to understand. Parang ganun yung idea doon. That's not supposed to be the case, di ba? Ang idea is, may pinaglalaanan ka, dapat. So dapat, it has to be understood, di ba? In a language that, yung accessibility ng knowledge itself has to be a factor in the way we communicate yung natin, yung output natin, be it either in published form or even in non-reporting. So yeah, I pretty much agree with that. Thank you very much for those inputs. I think yung question rin naman na to na could be extended to Sir Ian, Ma'am Lair, and Sir Din. Do you have additional inputs? At least, not necessarily siguro in the genomic studies of human population, but rather in, for example, in wildlife science.
I think ganun din yung sentiment ni Ma'am Cora. The Philippine endemic species, lalo na kapag kami nakikipag-coordinate with the DNR, we have to consider these as not just your specimens in the lab, but these are the national patrimony of the country. So it is incumbent upon the lab to be not just stewards, pero talagang caretakers talagang not just stewards and caretakers, but talagang guardians ng mga specimens na to, especially with the unpredictable infrastructure and environmental conditions here we have in the Philippines. And case in point, nagkaroon kami ng scenario, nagkaroon ng pandemic, nasira yung aming -80 freezer and all our precious samples there gone. So it's sad. It's a sad reality. But ganun talaga. And now we have to rethink how we're going to store our tissue samples. So nitrogen, baka yung sa nitrogen storage, baka mas mainam siya for long-term storage of our samples. Magkaroon ng mirror collection sa biobank at saka sa Natural History Museums. These are the things that we have to consider. These are pragmatic, practical issues, but we still have to deal with them because we are ultimately the stewards, the guardians, the caretakers of the national patrimony of the country.
And Sir Ian, thank you very much for that input. How about Sir Din and Ma'am Lair? Anything to add?
I think in my case, I think Sir Ian already mentioned everything. So I'll pass for this one.
Okay, sige, no problem. Ma'am Lair, may dadagdag kaya?
Not much, but siguro just sa case ng wildlife, why yung question ni Ma'am Cora kanina na why are we doing this? in my case, I've been doing it because I really love nature. I love wildlife, even if it's not my expertise. So going back lang siguro some motivations natin, no, on why we are doing these things, and that will help guide also our actions and decisions. Yun lang.
Okay, thank you very much, Ma'am Nair, for that input. Okay, now next question, ito naman mga specific naman to specific persons talaga. Okay, so for Dr. Din, here's the question. You said that past events can leave genetic imprints on present-day genetic diversity of various species. Can this offer any insight on how changes brought about by climate change would affect genetic diversity in the future?
Yeah, that's actually one of the things why I keep highlighting selection, because if we know something about selection at the moment, and we know how the environment, for instance, will change, then perhaps we can project or predict how species might be able to adapt for something. Okay, so definitely we can use those information, and that's actually one of the reason why genetics or genomics is very important in conservation at the moment. That's kind of like the holy grail for us evolutionary biology. What are the drivers of selection? What are the genetic basis of these selected traits? And will we be able to leverage them so that we can predict what will happen to this population? Will the population survive if the environment change to this direction, and so on? So there are some works, like for example, my supervisor, Cynthia Reginas, they did some work in coral, kind of predicting if coral will be able to disperse against climate change, something like that. So yeah, that's essentially the goal of one of the goals of genomics in conservation and management, understanding how selection work and using those information to help prepare a biodiversity for future climatic changes. Yeah.
Okay, thank you very much, Dr. Din, for that answer. Okay, now there's another question. This one is for Dr. Ian Fontanilla. Are you aware of any updates regarding the current status of the Earth by Genome project given the pandemic? It seems very unrealistic to uncover all of Earth's genomic diversity by 2028. Even studies on human diversity are very incomplete and focused largely on people of European descent.
Yes, definitely it's not realistic to cover all the eukaryotic multicellular organisms by 2028. But at least it provides an impetus to generate a snowball effect to include as many institutions and groups as possible. And that is why, at least from the perspective of the Philippines, we need to participate actively on this kasi hindi masyadong represented ng biodiversity ng Pilipinas in major database. It's quite ironic because we are a mega biodiversity hotspot. The pandemic has indeed had an effect on the progress of the EBP. I cannot speak on behalf of EBP because I'm not part of EBP, but definitely I think it has an effect on the progress, especially in many countries with regards to I think the supply chain pala at saka yung access to sites where you need to sample, particularly in those where they need to collect the taxa for their whole genome sequencing. And this will also be the case for the Philippines. Definitely hindi tayo makakasimula kagad na para bang para hitting the ground running kasi once mabigay man yung funding, we don't know what the status of the pandemic will be 1 year, 2 years down the road. But definitely it will have an effect on the pace of our research.
Thank you very
Dr.
much,
Yap,
Sir Ian.
unahin daw ni
Yes.
Dr. Yap, unahin daw ni Dr. Ian yung nasa pera. Yun ang sequence.
Actually, maganda nga yun kasi it's a good justification. Well, at least from the government perspective and in the mind of the public. Oo nga no, kasi we put these taxa in our Philippine notes sa ating currency. So and for good reason, whatever that reason may be. These are para bang charismatic, so to speak, charismatic taxa. and having whole genome sequencing them will probably will trigger something in the minds or imagination of the public. And foremost of these is of course the Philippine eagle, because the Philippine eagle is always associated with the Philippines. So yeah, it is para bang mahalaga siguro na basta as a national pride man lang kahit paano ma-sequence natin yan. And with that, talagang ibinabato ko yung responsibility sa sa UP Mindanao kay Doc Nair. So I hope ma-push po ito, ma'am.
I think puput pa rin naman ni Ma'am Blair yun. Di ba po? Yun. Okay. Thank you very much, Sir Ian, and also Ma'am Cora for the additional feedback. Okay. Okay, now there are some questions now in the Q&A tab. Let me see if we could get one. Okay, this is a very specific question. I'm not sure if you want to know. For Dr. Nair, how will you address the acquisition of agents problem that you're experiencing?
Ano nga
well,
ba?
oo nga
I always
no
say,
problem.
I always say good planning.
Mhm.
And for the most, it works. But
Mhm.
it's not always, it doesn't always work, especially now in the pandemic. So how I wish we have laws that are more forgiving and are more enabling when it comes to government procurement for research, for research. Sana may ganong...
So I guess pa kailan maglabi.
I totally agree with that, Ma'am Lair. That's why I mentioned a while ago that it's on the level of policymaker. I think because there's essentially some problem with our what they call that law, like something related to acquisition of material, like the bidding, etc.
9184, I think, is the law. I could be wrong.
And
Anyway,
may I speak pala? Kasi manood pa po tayo.
Dr.
Manood pa po
Ma'am.
tayo ng ating
Dr. Salama is also
meeting.
here. Okay.
So everybody, no, itong, because we have been, we are always beset with the problems of procurement. May mga developments na naman at may mga developments at may mga changes din naman. So for example, in many ways, UP Diliman for us is very supportive. UP system is also supportive. You can convince them. For example, sa Genome Center lang, I'll just share with you the story when the first time we procured our reagents and equipment with the first phase of the PGC, and this was in 2012. Sa palagay ko, mga 700 biddings yun, yung ginawa sa PGC first batch. One would be, yung iba, we went directly to the BOR so that we will have exemption from taxes. We were able to do that with 30 million equipment in lump. And for the others, what the SPMO of UP Diliman did was every week, mga 7 na mga materials ng PGC nakapalag, Diliman pa to, ha? Palagi nakapasok doon. So it is very important that you make engagements with the SPMO. At gumawa naman talaga ng special back ang Diliman for the buildings and also for technical. But sometimes, depending on the administration, sometimes that there are some changes, but they're actually amenable for you to improve. Si Pia nag-a-attack yan sa back. Si Pia, Dr. Pia, attack mode yan. So anyway, very good naman sila. Now, but there's also something very perplexing with this. We are governed with the same procurement law throughout the land, pero yung practice sa Philippine Carabao Center, yung practice sa DOST, at ang practice sa UP, iba-iba. Hindi ko nga sila maintindihan. Kung minsan sa iba, below 500 million, pwede na daw ganito. Bakit sa atin every time beyond 50,000 ganito, masakit na talaga yung ulo ko. So sabi naman ng DOS, gumawa daw sila ng bagong, totoo ba yan? Hindi ko binasa yung law, tungkol sa procurement that should be pro for us as researchers. Pero I don't know really if it's really making any big difference kasi we had some challenges in disbursing millions and millions and hundreds of millions of grants from the DOH and the DBM. The UP system is very, very supportive, particularly, and it happens every year. Baka ngayon hanggang December 31, the President, the VPLA, lahat-lahat, grabe kasi yung the threat is, pag hindi yan ma-approve at hindi yan anong tawag dito? Ma-award, ibabalik sa National Treasury. Palaging ganun, taon-taon. Now, nalaman ko naman na, ang problema sa UP Mindanao is really, really a big headache. And for many of our campuses within the University of the Philippines system, I think it's also a challenge to the local administration on how they're able to show that they can procure and support the researchers, particularly for multi-million projects, which are of national importance. No? So that has always been our problem. May mga proposals before. You remember, there was a time na parang sa US, parang naging warehouse yung laboratory mo, pag kukunin mo siya, at saka ka na magbayad kung baga nasa freezers mo na yung mga materials. But we have many rules about depending on cost. If it is 50,000, if it is 100,000, it is 500,000. We have many rules that govern this process. So hindi mo siya pwedeng kunin na lang diretso from the freezer. However, we should also learn from the experiences of other researchers. Ang ginawa nila ganito. Pag na-anticipate na talaga nila yung gagastusin nila, they always buy us a lot. They always buy us a lot. So for example, si Dr. Ray, na master na ng laboratory ni Dr. Ray ito because they've been receiving a lot of money. So the process in the beginning is a little bit tedious, but it's always based on luck. So maraming reagents, most likely one particular company can supply this. So isa lang ang bidding, one lot. So siguro yung mga bidding 5 lang. But that probably would be about 50 million of different kinds of reagents. So if you can help train your RAs to do that and anticipate bidding by lot, meaning to say ilump ninyo yung mga materials which are most likely pare-pareho yung maka-bid noon, mas madali. No? So that's going to be very easy. But there have been a lot of positive developments in the country in terms of grants compared to the time when we returned to the Philippines, where our annual budget was 50,000 to 75,000 pesos in MBB. So now we have students whose MBB BS thesis would be 1 million to 5 million. That's normal. But of course, we have some challenges. So for those who are still, this is a continuing struggle, but I think we can learn from the experiences of other researchers who are able to go it within the law, but be very creative and effective in terms of procurement. So yun lang ang masasabi ko. And ang problem ni UP Mindanao, hindi pa namin nasusolve. Ngayon, masakit ulo ni Dr. Lion na yan. Okay, yun lang, Dr. Jan.
Okay, thank you very much, Ma'am Saloma, for that comprehensive answer to the question. I suppose that would also help the audience, especially the one who asked the question. Maraming salamat po for your input and for joining in. Okay, sige. Move tayo sa next question. Meron pa tayong around 5 minutes to ask. Okay, so there's a question here. Okay, I think this is for Dr. Fontanilla and possibly for Ma'am Saloma rin and for other the bio and MBB. Will there be a curricular shift in the biology genetics degree program to mandatorily include bioinformatics and programming at least?
Yes, I think there's a push na. I think in NIMBB, kasama naman talaga, Ma'am Cynthia, yung bioinformatics. So,
True.
So maganda po yun na if you're from NIMBB and then you shift to or you pursue MS or PhD bioinformatics, it's not really a major shift kasi meron ng background. But at least for bioinformatics is an elective, and I think we have already teamed up with Sir John when he was still in the Department of Computer Science, wherein he taught bioinformatics as elective, and also for our graduate programs. Even until now, nagtuturo si Sir John. But at least for bio, it's definitely a major elective now. Sinasama na namin siya and we encourage people to participate. But at least for NIMTP, I know that bioinformatics.
Yeah, many years ago pa.
Yes, ma'am.
Can I add something, sir Ian? And also for bio. So for Bio 100, which is biostat, we're also introducing programming in biostatistics. And so that's one thing. And also we are overhauling some of the courses like biogeography. So there will be
Very good.
intensive kind of hands-on programming. It will involve programming to address some biogeographic questions.
Oo, Dr. Matias, feedback from MBB kasi binabantayan ko lahat ng estudyante ng MBB na kumukuha ng Bio 100, no? The offering in biology for Bio 100 is professor dependent. Ibig sabihin, kung ganito professor nagtuturo, ito ituturo. Pag may ganitong professor ituturo, ito ituturo. So probably we're very happy. We're very happy that we were able to recruit faculty members, and I think we should have some more consistency in terms of the course content.
We're trying to create like um, kind of like we're trying to standardize things, particularly
Yeah,
for
very
the
good, very good.
practical session. So we're
Yeah.
coming up with like modules for the practical
Yeah.
sessions that students
Tsaka
can work on.
dapat kahit sino nagtuturo, pareho pa rin ituturo. Kasi ngayon, nagdumating si Dr. Matias, dinagdagan niya, pero I have experienced many years, nagre-reklamo mga MBB sa course content. So Dr. Ian is familiar with this. And idagdag ko rin, Doctor, sa discussion ito, this is our third day seminar, last day of the seminar. Alam mo yung challenge with bioinformatics and incorporating genomics and bioinformatics in the way we teach our undergraduate as well as graduate curriculum is really, really a challenge. Hindi pa natin na-point out doon on how we teach, for example, genomics in the teaching of medicine, di ba? You remember that the technology is evolving so fast that sometimes we either would not have the expertise for teaching, or sometimes the curriculum takes a while to be taught. So we also would like to point out, for example, chemical engineering and biotechnology and molecular biology have undergone an enormous amount of changes, so much so that the traditional chemical engineering is really molecular biology. Because you're using molecular biology techniques, for example, for enzyme expression and purification. But sometimes because of lack of faculty, the traditional way by which it is, the way by which it is being taught is still the same. But that's why we need we need expertise. We need faculty members who are able to teach these courses. And number two, of course, we need to regularly overhaul our curriculum. And for areas where we don't have enough faculty, some universities can help other universities by creating modules which they can use to also convey this information to their students. And we have a lot of challenges in the Philippine higher education system, not only bioinformatics and genomics, even cell biology, the way cell biology is being taught, both in the lecture and iba pa yung facilities and access natin sa laboratory. No? Kasi mahal naman din talaga yung mga ganito. And of course, do we have faculty members who are ready to partake or to teach these courses? So, but at any rate, mga ginagawa natin na ito sa pandemic, particularly sa bioinformatics, kasi hindi pwede face-to-face classes, hindi pwede pa ngayon wet lab, talagang puros bioinformatics ang mga projects sa mga bata. At saka puros molecular modeling. No? But syempre for molecular biologists, gusto din naman nila matuto ng experiential kasi yung laboratory work, from pipetting to cloning to expression to purification. So hopefully we can weave both of these for those people who are really very good at computational problems. Maganda yun. At the same time, of course, we also need experiential way by which we can do this in the wet lab. So yun lang. Kami dito kasi tayo, we are national institutes, the national institutes have an obligation to capacitate other schools in these techniques, and the Philippine Genome Center takes our role also in this effort for genomics and bioinformatics very seriously, and we are always open to help different universities, colleges, and students get the information and the necessary skills as well as techniques in these emerging disciplines. So yun lang po, Dr. John.
Okay. Oops. Ayun, nag-timer. So, there's actually one last question here, but
no sound.
Dr. John, na-mute ka?
Oo, walang sound, Dr. John.
Hello po?
Na-mute ka. I think you hit the mute button.
Actually, okay, internet connection is unstable. So, can you hear me now?
Yes. Go ahead.
So, again, my internet connection became unstable again. So, apologies for that. There was one question actually in the Q&A asking for any available opportunities in your respective fields for interested people. But unfortunately, we've run out of time. Okay. So, can you still hear me at this point?
Yes.
Okay, right. So, unfortunately, I guess, ano na lang, kung sakali man, kung pwedeng, I would just like to request the speakers to provide some contact details or a website to visit for our audience members to check kung ano yung mga opportunities available for them. All right? So, paki-ano lang po sa chat Tapos,
At saka
addressed to everyone.
Dr. Yap, i-ano mo na rin, i-sell mo na rin na you remember we're creating modules for high school students no sa bioinformatics.
Yes,
And
ma'am.
we're starting
Actually,
off
na,
with
na po.
UPIS na. Okay.
Yes po. Nakaano ko na po. Yung GB Learn is actually part of that. That's yung public platform natin. Okay. So, sige. So, let's see. Okay, sige. With that, okay, I'm now going to close the Q&A portion of this program. Okay. And then, as a token, as a very simple token of appreciation, right, for your efforts, for the efforts of the speakers, for providing their expertise, time, and effort and input to the various topics under biodiversity, ethnicity, and forensics, we would like to provide. Okay, the Philippine Genome Center would like to award this certificate of appreciation to the speakers. I'm just gonna read the general details. So the Philippine Genome Center awards this certificate of appreciation for being a speaker during the Genomics and Bioinformatics Conference, Overcoming Challenges and Building Opportunities on December 9, 2021, hosted from A. Maria Regidor Street, PGC Building, University of the Philippines, Diliman, Quezon City, 1101, signed by Executive Director Dr. Cynthia P. Saloma, and yours truly as the Program Director of Core Facility for Bioinformatics and Head of the Program Committee. So first, we're awarding this to Dr. Maria Corazon de Ongria for being the keynote speaker for today's proceedings. So ma'am, ito po kunwari, binibigay ko. So thank you very much, ma'am, for coming and for sharing your expertise and insights. Okay, next up, this certificate is awarded to Mr. Frederick Delphine, C. Delphine, for being a resource/plenary speaker. So, Sir Fred, okay, thank you very much for coming and for accepting the invite. Next up, you have Dr. Ambrosio Melvin E. Matias for being a resource/plenary speaker. Din, maraming salamat sa pagdalo.
Thanks for inviting.
Yes. Next up, we have Dr. Lar Ani E. Moral, okay, for being a resource/plenary speaker. Ma'am, maraming salamat po. And last but definitely not the least, Dr. Ian Kendrick C. Fontanilla for being a resource and plenary speaker. So po, Sir Ian, maraming salamat ulit sa ulitin po. Okay? Yeah. All right. So, that said, I would like to request yung for a picture taking or photo op muna before we So, Rachel, you still there?
Hi po.
Okay.
Good
All right.
afternoon po. We'll be taking the screenshot po.
Okay, sige. All right. So, ako na mag-countdown?
Wait lang po, sir. Dr. Distura, if you can join po, sir.
Yes, Dr. Distura, please. Sir.
Open your camera.
Sir, there.
Yeah, I'm here. Can you see me?
Yes,
sir.
yes.
Alright po, I'll be counting po. Ready? 1, 2, 3. Alright, saving lang po and then one more shot. Taking the photo. 1, 2, 3. Alright, thank you po.
Okay. Thank you very much to our distinguished speakers and thank you very much for accepting the invite. Sa ulitin po. Okay? Now, to formally close this program, we have our Deputy Executive Director of the UP Philippine Genome Center and one of our esteemed celebrities from the center as well, Dr. Raul V. Destura, to give the closing remarks. So, Sir Raul, the floor is now yours.
Yeah, thank you so much, Dr. Yap, for the opportunity to close the conference. You may have all our audience may have already appreciated the importance and the roles genomics and bioinformatics have played over the years, especially during the time of the pandemic we're in. The role of genomics and bioinformatics has always been integral in the monitoring, surveillance, and opportunities for determining risk factors in the world of healthcare technology development, in the world of vaccine development, in the world of therapeutics development, and public health monitoring. And that's just for health implementations and health applications of the genomics and bioinformatics uh, pool of knowledge. But you have seen over the conference that it expands beyond healthcare, also in the area of biodiversity, ethnicities, and all the other aspects related to the art and science of genomics and bioinformatics. Truly, the efforts of our dedicated scientists and bioinformaticians in the Philippines have contributed significantly to the growth and diversity of the science, genomics, and bioinformatics in the country. The Philippine Genome Center and all the organizations and scientific community in providing good science, good evidence, and good areas for which generation new knowledge through the art and sciences and bioinformatics. Making this conference truly a huge success and looking forward for everyone's session and webinars at the Philippine Genome Center. I now formally close this conference. Thank you very much and have a wonderful day.
All right. Thank you very much, Sir Raul. Actually, we also have something as appreciation for your taking the time. I know you're very busy with all the things happening around. Okay, guys, yung ano natin. We would like to also extend or award this certificate of appreciation for formally closing this event. So thank you very much, Dr. Destura. Okay, mabuhay po kayo. Okay, and with that, okay, comes the end of the GBCon 2021 Genomics and Bioinformatics Conference, Overcoming Challenges, Building Opportunities. There's a feedback form, okay, that to help us assess how we did, okay, and this survey form will also serve as reference for the registrants' attendance to the conference. This form will be open until 6 p.m. December 9, 2021. Just make sure to type in your full name correctly, as certificates of attendance will be generated based on the form response. Okay, so it's also that the link can be found in the chat box. All right. Okay, so with that, GBCon 2021 is over. Okay, thank you very much. We hope to see you again in GBCon 2022 Electric Boogaloo, I think. But maybe I'm wrong. But still, ingat palagi, stay safe, stay healthy, and thank you very much for coming. This is John Michael Yap signing off.
Thank you very much. Thank you.
Thank you very much, Sir Ian, Ma'am Lair, Sir Fred,
And
Sir Din.
yeah,
Thank
thank you very
you.
much to the
Ma'am Cora.
team of Dr. John and to everyone in the organizing committee. Thank you very much, and to our speakers. Great talk this week.
Congratulations, everyone.
Thank you.
Congratulations, everyone. Take
Okay.
care.
Bye.
Okay.
Thank you po, speakers and attendees.
Okay. Thank you. Thank you, everyone.
Is a multidisciplinary unit in the University of the Philippines system, providing research, training, and sequencing and bioinformatics services. For over a decade, PGC has been leading the country's research and development drive in genomics and omics-driven technologies. Initially envisioned as a facility which will contribute to a timely and strategic response to threats of emerging infections, the unfolding events in 2020 prompted the Genome Center to utilize and optimize its resources to address the unprecedented challenges posed by the coronavirus pandemic. With its recently established clinical genomics laboratory, PGC equipped and reconfigured the lab to comply with the requirement of the Health Facilities and Services Regulatory Bureau of the Department of Health, and was certified to conduct testing for COVID-19 by real-time PCR. In coordination with the Department of Health, the COVID-19 testing lab started off as a non-hospital based facility which accepted samples of priority cases for testing from hospitals, community swabbing centers, and other healthcare facilities in Manila and nearby cities and municipalities. To address the growing number of cases and contribute to targeted mass testing, PGC's Clinical Genomics Lab expanded its screening operations by opening a drive-through and on-foot swabbing area in the grounds of the Institute of Mathematics. In addition to its testing efforts, PGC fully utilized its DNA sequencing and bioinformatics core facilities in identifying nearly complete whole genome sequences of 6 SARS-CoV-2 from COVID-19 cases between March 22 to 28, 2020. It has since serviced the requests for next-generation sequencing of SARS-CoV-2 from other referral hospitals in the metro. To provide a broader view regarding the sources, spread, and evolution of COVID-19 locally, PGC is spearheading a national effort to do genomic biosurveillance of SARS-CoV-2 in the country. As the only local facility with a range of next-generation sequencers for a large-scale sequencing effort. Hand in hand with PGC Visayas and PGC Mindanao, NIH in UP Manila, and NIMBB in UP Diliman, the satellite facilities of PGC in Iloilo and Davao City have conducted trainings in molecular biology techniques covering RNA extraction and RT-PCR for COVID-19 testing in the regions. These efforts are particularly important due to the constraints in inter-island travel at the height of the pandemic. The trainings have been a boon to local laboratories, centers, and hospitals as they prepare to be accredited as COVID-19 testing laboratories in their specific localities. These trainings have contributed immensely to expand the country's testing capacity at a critical juncture in our nation's fight against the COVID-19 pandemic. The Philippine Genome Center continues to advance its position as an institution on relevant multi-omics research and services that responds to the needs of the Filipino people, utilizing genomics for a better Philippines. Ang PGC para sa bayan.